dar inicio a la tercera sesión del simposio, y para mí es un gran gusto de presentar al primer ponente de esta mañana, al doctor Carlos Velázquez Martínez. El doctor Carlos Velázquez realizó sus estudios de licenciatura en la Universidad de Guadalajara y posteriormente sus estudios de maestría en química farmacéutica bajo la dirección del doctor Rafael Castillo. Posteriormente realizó sus estudios de doctorado en la Universidad de Alberta, en Canadá, y una estancia postdoctoral también en el área de química farmacéutica en el Instituto Nacional de Cáncer en Estados Unidos. En el año 2008 se unió a la Universidad de Alberta como profesor en la Facultad de Ciencias Farmacéuticas, donde actualmente es full professor. Sus líneas de investigación se centran en inhibidores del factor de transcripción FOXM1 en dos áreas, en moléculas pequeñas que se unen directamente a FOXM1 y en Protac, que como el doctor Carlos lo va a presentar, verán que es una de las tendencias actuales en el descubrimiento y diseño de fármacos. A lo largo de su trayectoria como profesor en la Universidad de Alberta ha recibido numerosos research grants, proyectos de financiamiento, sumando poquito más de 2.1 millones de dólares. Ha dirigido y dirige varias estancias postdoctorales, 6 en total, 9 estudiantes graduados, de posgrado, 14 estudiantes de licenciatura y también ha recibido a 7 alumnos en estancias de investigación. Cuenta con más de 60 publicaciones en revistas indizadas, destacando que 40 de las publicaciones más recientes tienen una proporción de prácticamente 18 citas por artículo, con un H-index en total de 16. Ha realizado y ha participado en 2 patentes y es un gran gusto para nosotros recibirlo. Muchísimas gracias, doctor.
Muy buenos días a todos. Una nota muy rápida. Mi buen amigo Alfonso me pidió que hiciera la plática en inglés por cortesía al invitado que tenemos el día de hoy. Entonces voy a dirigirme a ustedes en inglés. No crean que se me haya subido lo sangrón. Para efectos de que el invitado nos entienda, entonces vamos a comenzar en inglés. Good morning, everybody. Welcome, professors. Welcome, everybody. It's a great pleasure for me to address this symposium in honor of a great mentor and a great friend, Dr. Rafael Castillo, as well as Dr. Alicia Hernández, Dr. Alfonso Lira, Francisco Hernández, Sergio Rodríguez. The list is just endless. Thank you all for having me. First things first, because I'm going to talk to you in English, I need to make sure you understand what I'm saying. I need to make sure that you guys are not falling asleep. So, I hope this thing works. Okay, let me see. Retry. I should have checked. I was trying to make this interactive, but because I may ask you to scan a QR code later on, so that I'm going to ask you some questions. All my presentations and all my seminars are interactive, because otherwise, after 30 minutes, you start checking your Facebook page or your Twitter or X, whatever the case may be. So let's get started. Where are we? The University of Alberta is located in the province of Alberta in the city of Edmonton. If you guys ever want to visit me, it's just only 4,822 kilometers. It's just, or take a plane. It's up to you. This is the city of Edmonton at night. This is how it looks like in summer. And it's a beautiful country, Canada. It has a lot of hiking, biking, canoeing, rafting for the adventurers. If you are feeling like you like the adventure, please go there in summer and visit. Unfortunately for us, Canada decided to implement the visa again, but nonetheless, this is some of the things that you may look and you may see if you decide to go backpacking, if you decide to go camping. At the beginning, when I was going to Canada, I thought this was just something out of a picture of a photographer, but believe me, it's like that, or better. It looks like it. Then in fall, trees start changing, the chlorophyll starts undergoing changes, and then all vegetation becomes yellow all of a sudden. But then winter comes, and then we have temperatures up to minus 30, minus 35, at which the moisture on our eyes, on our ears, on our cheeks, the face starts freezing. So it gets like that. It's not the idyllic paradise. It has its own disadvantages, but nonetheless, it's worth visiting. This is the Cates Building for Pharmacy and Health Research. This is where I conduct my academic and professional activities. This is how it looks like, and this is where we are starting to work with the FOXM1 protein. Let me just give you a little bit of a background in order for you to understand what I'm talking about. The FOXM1 protein is a member of, let me see if I have a laser pointer. Yeah. FOXM1 protein, it's a member of a much larger family of transcription factors that has more than 16 different variables, and each one has its own subtypes. FOXM1 proteins are involved in a wide variety of biological processes associated with DNA replication and cell cycle control. So in other words, FOXM1 is involved in cell replication and cell division. However, despite that we need FOXM1 for normal metabolism, sometimes normal or aberrant activation of receptor tyrosine kinases, more specifically RAS, RAF, or MAPK2, results in malignant phenotypes. In other words, when cancer appears and when things start to go wrong, okay? Accelerated ERK-mediated FOXM1 phosphorylation increases nuclear accumulation and activation of dependent transcriptional cascade. So if you can imagine, whenever the cell needs to replicate itself, FOXM1 is phosphorylated, it's translocated to the nucleus, it binds to DNA, and it starts the transcriptional cascade that ultimately leads to cell replication. You can guess, cancer cells have learned to overuse this process in order to replicate faster, okay? Therefore, whenever FOXM1 is present and it's overexpressed, you see a whole variety of other proteins that are also overexpressed, such as superoxide dismutase 2, it's cyclin A1, B1, survivin, BCL2, VGF, and so on. The list, this is just a small list of proteins that are activated after FOXM1 is phosphorylated. What is the net result of this? Well, patients start developing chemoresistance, and there's a poor prognosis. Those cancer types that are expressing FOXM1 in high amounts are the ones that have the poorest prognosis. Many years ago, medicinal chemists learned this was an important drug target. Therefore, there has been a wide variety of pharmacological treatments, including peptides, small molecule drugs, and siRNA. All of them, when they inhibit FOXM1, the net result is this. There is decreased cancer cell migration, cancer cell invasion, decreased metastasis, and decreased cancer stem cell inhibition, or cancer stem cell inhibition. As you can guess by now, whenever we inhibit FOXM1, there are many different pharmacological desirable properties that we are looking forward to. As you already guessed, because FOXM1 is a transcription factor, transcription factors are notoriously difficult to develop targeted or therapies against. Consequently, there has been a wide variety of publications reporting FOXM1 and 4 different strategies. Strategy number 1 is inhibition of protein-protein interactions. Let me explain what I mean by that real quickly. Imagine FOXM1 on one hand, another protein that promotes the binding of FOXM1 to form a dimer, and then it binds to DNA. The second strategy involves targeting the transcription factor DNA binding domain. So if you can see FOXM1 as my hand here, and whenever it sees DNA, it grabs DNA and latches to it. So if we inhibit those DNA binding domains, we release the DNA and FOXM1 goes away. The third strategy that you see there is the chromatin remodeling or targeting the epigenetic readers. So in other words, epigenetic modifications need to take place in order for FOXM1 or any other transcription factor to bind to DNA. Therefore, if we inhibit chromatin remodeling, we inhibit its binding to DNA. Or using transcription factor decoys. In other words, imagine FOXM1 as my right hand and a decoy as my left hand. If this decoy resembles or looks like FOXM1, it can grab the DNA and get it sequestered. Therefore, DNA remains free. So that's the fourth strategy. The developing of Protacs that I'm going to talk to you about today involves the second strategy, targeting the transcription factor DNA binding domain. You can see here the long list of cancers that overexpress FOXM1. And whenever this happens, bad things and bad prognosis, chemo resistance, and a lot of other negative consequences. FOXM1, it's overexpressed in most, if not all, cancer types. And this is why it's so important to develop new therapies against it. And this is where, let me see, more than one window. Please close all the windows. I was trying to make this interactive, but I may have to save that for other day. Let me continue with a little bit of the background. I'm not going to talk to you today about my other project. I thought this was going to be a lot more interesting to you. Some of these data is the first time I present, so it's fresh out of the oven. What are Protacs, or proteolysis targeting chimeras? I'm not going to read this to you, but the most important thing is that imagine Protacs as bivalent molecules. Imagine one drug in one hand and another drug in another hand, and then a linker in the middle. Okay? One of those medications, or one of those drugs, is responsible for binding to the protein of interest. The other drug is responsible for binding to an E3 ligase. This is very important. Please remember it. I may ask you guys after, especially if I see you sleeping or checking your phone. So you better pay attention to this because it's very important. So just to recap, one drug binds to, in this case, FOXM1. The other drug binds to an E3 ligase and then a linker in the middle. This is very important. Why? Why would we need two drugs, two different pharmacological compounds here? because whenever this E3 ligase is engaged, the proteasome recognizes that protein and it breaks it down. Imagine yourselves going through normal metabolism. Whenever we produce proteins for our tissues, our organs, whenever the catabolism is involved, those proteins that are no longer needed in our metabolism have to be broken down, have to be metabolized. The E3 ligase proteasome system is responsible for breaking down normally, naturally, those proteins that we no longer need. However, when we hijack this system and we artificially induce this breaking down, we are promoting the degradation, therefore the name. We are promoting the degradation of whatever protein of interest we are examining. And this is exactly what I was just talking to you about. Imagine a polypeptide called ubiquitin. It's blue circles that you see here. So the first stage is the activation stage. An E1 ligase, it's engaged, it's ubiquitinated, then in the conjugation stage, another E2 ligase, it's also involved. This E2 ligase peptide, it then associates with an E3 ligase, and this is the net result. This is the substrate or the protein that needs to be degraded. And this long chain of blue circles, it's called polyubiquitination. It's a tag. I'm doing something wrong here. This is like a tag. We tag this protein and then we mark it for degradation. Please note that this is different, totally opposite to regular protein inhibition. We are talking about not inhibition, not binding or irreversible binding. We are talking about breaking it down, okay? Then the proteasome is involved. It recognizes this tag. This is called proteolysis. And then the protein that is causing trouble is degraded. It's effectively just discharged, just like any other protein. And this is what a Protac looks like. So imagine one drug on one hand, the other drug on the left-hand side. This is the protein of interest in blue. This is the E3 ligase. And when the Protac engages both of them, then this happens. The ubiquitination takes place. And whenever this takes place, that protein will be degraded. The proteasome just releases ubiquitin, and then the protein is just releasing in single amino acids. Quick question for you, because I told you this is going to be interactive, so you guys don't fall asleep, and I need to make sure you're following me. What do you think this model be compared to classical enzyme or protein inhibition? What are some of the advantages and disadvantages that you can think of when designing drugs in this fashion? Anyone? Real quickly. Anyone here? Yeah, go ahead. High specificity, yes, it depends on the specificity of the drug that binds to your protein of interest, right? Yes, what else? Let me see, who's using his phone here? Yeah, all of a sudden, magically, no one's using the phone. Anyone in a red blouse?
You showed us that you have different FOX protein. Maybe they can bind this.
Yes.
And they will destroy it.
Yes, so they're different isoforms, yes. So depend the selectivity, the Protac will be as good as the selectivity of your drug, right? Now, what do you think about this being cyclic? What does that tell you? What does that tell you? What does that mean? That it's cyclic. It goes around and around and around. What about the stoichiometry that we need for the Protac, it goes around and around and around several times, several cycles until the Protac is metabolized itself. But by then, one molecule of Protac has degraded several molecules of the protein of interest, right? This is perhaps the biggest advantage of the Protacs. This is how Protacs look like, a little bit more specific. This is some of the target proteins that have been engaged with Protacs. BRD4, the androgen receptor, BTK, CDK, several linkers, and then the E3 ligases. The drugs that we use to engage E3 ligases are several. The list goes on and on and on. These are some of them. Some substrates for many types of E3 ligases that we have in the body require different substrate. Consequently speaking, if we know the type of E3 ligase that is expressed in the type of tumor that we want to engage, then we use that substrate and we increase the specificity. And this is how a full Protac looks like. So this is one molecule on one hand, the linker, and then the other molecule on the other side. So that's how Protacs look like. All right, this is not gonna work. A history of pioneering. Perhaps the company that is most advanced in this technology is Arvinas, and they started this concept just about the time I was starting my PhD degree in Canada, around the year 2001. And then by the year 2004, they have the first phase 1 trial with Protac for neurodegenerative diseases. This is the other advantage. Think about any disease. Think about any protein that you have in your projects. If you have a drug that binds to that protein anywhere, doesn't matter how, if it binds to that protein and then it engages the ubiquitin proteasome system, you have the ability to develop a Protac against that target. Okay? This is how Protacs are divided. This is very interesting. If I can have your attention here for just a little bit, I will appreciate it. This is the concept that I just described to you, Protacs. The E3 ligase, the protein of interest, and then the Protac in the middle. You can see between them, it's like a sandwich. It degrades that target, gone. But there are also another type of degraders described in the literature that are even more interesting from a computational point of view. I know here there are many researchers really good at computational modeling. These are called molecular glues because these are a lot simpler, a lot smaller, a lot lighter. Molecular glues effectively do exactly the same thing as a Protac, but they're smaller and they don't require a linker. You can see here how the net result in both cases is the activation of the ubiquitin proteasome system, right? These are the two distinctions of molecular degraders. Molecular glues on one hand and Protacs on the other one, the classic ones. Many drugs could be molecular glues. So one advice I have for every researcher here, think about this when developing drugs, because they could be degrading proteins, and we don't even know about it. So this is just a general introduction. Now let me show you what I'm doing in my lab and what are some of the results we have obtained with some of this concept. I'm going to divide my talk in two parts. Part number one, or part three in this case, is the work that we've done with triple-negative breast cancer cells. This was the proof of concept. And then the more recent one, our work with Protacs as a potential treatment for high-grade serous carcinoma or ovarian cancer. This is ongoing work. We started about three and a half years ago with very basic, very rudimentary molecular modeling and molecular dynamic simulations just to make sure that the Protacs that we were trying to design were going to fit in the interface between the two proteins. And just so that you imagine what this is like, imagine one big protein on one side, the other protein on the other side, and then the small molecule, compared to the size of the molecules, despite Protacs being relatively big, they are relatively small comparing with the size of the two proteins, right? So we needed to make sure. Question for you, question for everybody here. What happens if the length of the Protac is too short? The two proteins will approach one another too much, and they start repelling each other. What happens if the length of the Protac is too big? They're too far away, they don't engage. So part of the design and the developing and the concept is to find that sweet spot in the middle. Not too long, not too small, right? So this is some of the things that we've done in terms of the chemical synthesis. I'm not gonna stop here for too long, but just so that you know, this is the E3 ligase ligand, compound number 13, thalidomide, and through a series of synthetic steps, we ended up with a series of Protacs. The drug that you see on the left-hand side here is the E3 ligase ligand. This is the linker in the middle. And the molecule you see on the right-hand side is the FOXM1 inhibitor. I'm going to have some questions for you here. These assays are called the electromobility shift assay. And if you can follow what I'm saying here for a minute, I will appreciate it. Remember that I told you that the Protac need to bind to the DNA binding domain, right? So imagine the drug alone, okay? Imagine the DNA binding domain, not the entire protein, just the sequence of peptides that are responsible for binding to DNA. That's what we call the DNA binding domain. Just a sequence of small polypeptides. This is how it looks like. Imagine you put this on the top. This is a gel. You apply with a pipette on the top of the gel, and then you run it through electrophoresis, so it runs down. So when the DNA probe is alone, it runs from the top to the bottom. This is how it looks like. However, when the DNA is associated with the FOXM1 protein, it becomes heavier. It's a lot heavier. So when we start applying increasing concentrations of FOXM1, remember this is just DNA and this is FOXM1. What happens? As we increase the concentration of that polypeptide, more and more FOXM1 starts binding to more and more DNA. It becomes heavier and heavier. The band at the bottom starts disappearing, and the band at the top starts appearing. This is how we know that FOXM1 is associating to DNA. This is what happens normally. So, question for you. Let me see, I need a brave volunteer with a beautiful voice here, just to make sure. Anyone with a yellow blouse? Anyone with a yellow blouse? I need to make sure you guys are understanding me and you guys are following what I'm saying. Raise your hand if you have a yellow blouse. No. Resulta que nadie tiene blusa amarilla. ¿Cómo se llama, amiga? ¿En medio? ¿Ana Laura? Alondra. Alondra. Mucho gusto, Alondra. Nice to meet you, Alondra. What do you think it will happen if we start adding drug here to this phenomenon? So remember, FOXM1 is on one hand, DNA is on the other. And when we put them together, they bind. What do you expect to happen if the drug is active? It starts disrupting that association, right? So we start seeing each one individually. And this is what you see here on the right-hand side. You see here, when the DNA probe alone is used as a control on the left-hand side, and this is where we start. This is the associated protein to DNA, and look what happens when we increase the concentration of drug. they start dissociating. It comes down again because it's lighter, right? So this is important because then we can build IC50 values and then we can see how active the compound is, right? Okay, this is how one of the Protacs looks like. This is compound 17D. Outstanding, thank you so much. You're the next one in my list of questions. So this is how the Protac looks like, and I want you to notice one thing very quickly. When the R group is equal to a methyl group, when it's methylated, the E3 ligase ligand is no longer active. Therefore, as a negative control, we methylate the R group here on this nitrogen. Whenever that nitrogen is methylated, it loses the ability to bind to E3 ligase. So therefore, it's negative control. Look at what happens to the concentration of FOXM1 when we use 17D, the Protac, at 5 and 10 micromolar. Look at what happens to FOXM1. This is the control band. It looks beautiful, dark, and then as we increase the concentration of the drug, it disappears. It starts to be degraded. Now look at what happens to the negative control. So it works. The negative control doesn't bind, doesn't engage to the ubiquitin proteasome system. We know it's working, right? All right. Now, this was done in triple negative breast cancer cells, MBA, MB 231 cells. Please pay attention, because you never know when I'm going to start asking you questions again. Okay. This is how the negative control in the Protac looks like. This is how compared to FDI6. This molecule you see on the left-hand side, right-hand side, here, is the FOXM1 inhibitor, okay? It's called FDI6. FDI6, it's a small molecule FOXM1 inhibitor. So we would expect to see inhibition of FOXM1 as we see it here. But what happens with the Protac is not that visible with this, but once we plot it in graph bars, then you start seeing some significant differences. So this is the vehicle when there is no drug in the media, cells produce FOXM1, and then when we add the FDI6, we see a decrease in FOXM1 expression. However, compared to the small molecule inhibitor, 17D, it's a lot more active. Same can be said for downstream modulators. So imagine whenever FOXM1 is expressed, it binds to DNA, it starts its transcriptional cascade, and it produces a lot more proteins. Some of those proteins are these ones. Okay, CDC25B, cyclic B1, and this is the control protein. So we would expect to see a decrease in the downstream modulators of FOXM1, which is exactly what happens. So if you see these bars, this is the concentration of FOXM1 in cells, and these are two of these downstream proteins that are also inhibited. Now, another thing that we did in collaboration with a Chinese researcher in China, they helped us a lot with these biological assays, is something called cell colony formation assay. This is one of the ways we can measure the anti-cancer activity of drugs. If you can imagine every spot, every purple spot that you see there, it's a colony that results, just like bacteria. You all remember the practices. I think you still, do you have bacteriology in yeah, some of you have. So just like any colony of bacteria starts from one cell here as well. So it starts from one cell of cancer cell in this case. So it starts growing, and every dot represents one colony of cancers. So you can see there the significant decrease in colony formation when we have 17D, and once again, the negative control, it's totally inactive. You can see it here. we see some decrease, but not as significant, right? And this is how it looks like. Interestingly, when we administer this to rats, imagine the poor rat, they inject a number of cells in the thigh, and those cells start growing, and they develop a tumor. Those tumors are isolated after a given number of days, and then those tumors are dissected, isolated, weighed, and measured. So this is how the tumors look like when there's vehicle, when there is no drug. When we give FDI6, there's a significant decrease, okay? And then with 17D, it's even more so. You can see it on the graph bars. This represents tumor mass. Tumor mass goes down significantly all the way to the Protac 17D, and we measure the tumor volume. Then you see here how there's a significant difference between the vehicle control, FDI6, and 17D. You see it there. It's a significant anti-cancer effect. Another assay that we received help from this Chinese group is the staining. So when cancer cells are stained or coloreadas, they start looking like this. So the darker the stain, the higher the concentration of FOXM1. So effectively, what we are looking for here is a decrease in the color or the intensity of the color. So you can see there the proof of concept developing the first ever reported FOXM1 inhibitor following the Protac approach. And then let me show you what we've done on ovarian cancers. Ovarian cancer is, specifically speaking about high-grade serous carcinoma, is the deadliest gynecological malignancy, which accounts for most cases and deaths in ovarian cancer. 75% of all diagnoses are in advanced stages, regrettably. Most patients respond well. They respond really well to the frontline therapy. But the big problem here is that after a few months or a few years, a significant number of those patients develop ovarian cancer again or some type of metastatic variant. And that's when problems start to come up, because then they are resistant to practically everything we have in the clinic. So this is why we are interested in developing a Protac that can attack this particular type of cancer. Why FOXM1? Why we believe FOXM1 is an important target in this variant of cancer? After TP53, FOXM1 is the most prevalent genetic variant in ovarian cancer. 84% of all patients in clinic have overexpressed FOXM1 in advanced stages. So therefore, if we developed a therapy against FOXM1, 84% of those patients will receive the benefit of it. This is the series of Protacs that we've synthesized, 96 in total. As you can see, I only have a limited amount of time with you guys this morning, so I can't present all the results to you, but these are some of the variables. So if you look at the left-hand side, we have 1, 2, 3, 4 different types of FOXM1 inhibitors. So in other words, the proof of concept that we developed with breast cancer now has to be fine-tuned in order for us to come to that sweet spot when we have several types of FOXM1 inhibitors to see which one is the best. Then we have to do some type of structure-activity relationship to find out what is the ideal linker length, okay? And then we also have to try it with several E3 ligands, thalidomide, lenalidomide, and pomalidomide. All of these drugs engage with a type of E3 ligase target called cereblon. The other type is VHL032, which is another type of E3 ligase. Both of these ligases are expressed in ovarian cancer, but we want to see which of those two yields the best result. All right? These are some of the Protacs that we have developed, much simpler, much shorter, much condensed, because here the goal, we know that this works, but now we want to do proper structure-activity relationships. And we start that with simple molecules. So these three drugs that you see here are exactly the same, except for the length of the linker. We have 1, 2, 3, 4, 5, C5 linker, C7, and C9. We want to see which length gives the best results. This is the FOXM1 inhibitor, and this is the E3 ligand. These are some preliminary results that I've never shown this before, so you guys are lucky. This is the first time we disclose this. This is probably the most aggressive type of ovarian cancer cell line, the OVCAR4. And what you're looking at here, let me see, is just a regular, very simplistic cell viability inhibition assay. So what you're looking at here is 3 different Protacs compared to the FOXM1 inhibitor that we also develop in the lab. I don't have time to go through it, but this is one type of FOXM1 inhibitor, TFI10. So we have the lead molecule or the parent molecule and 3 different Protacs, right? Now, let's see, another volunteer, another checkpoint. I need another volunteer just to make sure that you guys are following me. In English, I need one of you to tell me which of those 3 Protacs is the most active one. 18? The C9, yes, exactly. Yeah. And then, which is the next one? Yes. Yes, exactly. So as you can see here, we are also plotting the IC50 value in micromolar. Now, in this case, please note the big difference in between the initial FOXM1 inhibitor, which requires 1175 micromolar, compared to 0.06, 0.16, and 0.01. Now we're talking about low nanomolar range. This is the magic of Protacs, how they can increase the activity. So whatever the project you have, whatever the protein you're trying to target, you can start thinking about developing a Protac if you have a good ligand, because it can increase significantly by 1 order of magnitude or more the activity, right? Now, it's capable of inhibiting the pathological variant of ovarian cancer. What about normal ovarian cells? If you can see here, this is 72 hours post-treatment, and this is how it looks, the activity of those same Protacs, of those same drugs on normal cells. So you can see there is, they almost remain at about between 80 and 90% cell viability, and it remained constant. So it's not going down. It doesn't get any stronger. So we are achieving certain level of selectivity towards the pathological variant of FOXM1. And what I'm showing you here is the concentration-dependent activity of these Protacs. So compare Protac 15, 12, and 18. There is one feature here that I want you to please notice. I want you to please look at that. This Western blot on the top, and tell me if you see anything weird, anything different, anything that is just misplaced or out of the ordinary. Anything at all? No? No one? Yes. So please follow me here. Look at what happens with Protac 15 at 1 micromolar. Compared to the DMSO control, the activity goes up. So in other words, the concentration of FOXM1 is decreased. You agree? Raise your hand if you agree. Okay, no disagreements, good. What happens at 5 micromolar? It's more active because there is less FOXM1. Then something weird happens. Look at what happens to 20 and 40 micromolar. 20 and 40 micromolar. What happened there? What do you see? FOXM1 starts appearing again. That is called a hook effect in Protacs. The hook effect relates to this. Imagine the Protac and the protein of interest and the E3 ligase. So there's 3 entities there. The drug, the protein of interest, in this case FOXM1, and the E3 ligase, right? In order for this to be effective, the 3 of them need to be associated, linked, right? However, it has been observed that after increasing concentrations of the Protac, this starts going down. This trimer starts dissociating because then the dimer is favored. When the Protac is either linked to the protein of interest but not the E3 ligase, or the drug binds to the E3 ligase but not the protein of interest at increasing concentrations. So this is characteristic of Protacs, the hook effect. And this happens again here. So you can see at 1 micromolar, 520, there is practically no FOXM1, and then at 40 micromolar, it starts appearing again, right? Now, what happens with Protac 18? This is probably the most active Protac we have, because even at 40 micromolar, we start just a little bit of, the protein starts appearing again, but this is probably a very good sign. There's essentially no FOXM1 there. It's all been degraded, right? And then we started measuring also p-FOXM1. Do you know what p-FOXM1 is? I told you at the beginning. I need a brave volunteer. Let me see. Someone using the phone here? No, no one using the phone. See how beautiful it is when you guys are not checking your phone? And this event? All right, someone from the back. I need a volunteer. How about the last student on the right corner there? What's your name? Maciel. Maciel. Okay. What is that p-FOXM1? Do you remember? No? How about your friend next one? How about the next one? Dianita, don't fail me. I told you at the very beginning of this presentation, remember, in order for FOXM1 to be active, it needs to be phosphorylated. Once it is phosphorylated, it translocates to the nucleus. So what we are looking at here is the amount of phosphorylated FOXM1. And this is what we're trying to see the effect. It's not as pronounced because one simple reason. When FOXM1 or any other transcription factor is phosphorylated, it moves from the cell cytoplasm to the nucleus. And when it moves to the nucleus, the drug has problems crossing 2 membranes, the outer membrane of the cell and then another membrane of the nucleus. So it has double barrier. So the activity starts going down a little bit because now we are trying to degrade a protein that is phosphorylated inside the cell nucleus. You see? You see how much time we have? I'm running out of time here. I don't want to use Agustine's time here. So, but just to show you real simple how the concentration-dependent effect on downstream modulators of FOXM1, like CCNB1, PLK1, and Aurora B kinase, the effects are not as pronounced, but nonetheless, we see some effects. For example, in polo-like kinase, we have a really good pronounced effect with Protac 18. This is probably our lead Protac so far for ovarian cancer, Protac 18. Really quickly, often when I submit grants to get money, and this is something that I want every student here to please think about. Everything we do in the lab has a cost. It means money. Every time we open a reagent bottle, it means money. Every time we use solvent, every time we use a Western blot, every time we turn on the switch on the wall, everything costs money. So we have to be competing for grants and for money all the time. If we run out of money, we run out of everything. So whenever I submit for grants, this is one of the most consistent observations I get. Okay. You have a good FOXM1 degrader, but we know that there are other FOX proteins in the cell. How about the selectivity towards other FOX proteins? Well, you can see other FOX proteins, FOXA1, FOXK2, FOXO3A. You can see how the Protac 18 starts degrading some of them. Question for you. Let me select another volunteer really quickly. I'm going to finish my talk because I'm running out of time here. Quick question for you. How do we know which FOX proteins we want to degrade, and how do we know which proteins we don't want to degrade? Any ideas? Any? Let me grab another volunteer. Anyone with a black shirt with those fancy letters here of Symposium? Anyone? How about you? What's your name? Fernando. What do you think about that? How would you know if you were going to start this project, how would you know which FOX proteins you want to degrade and which ones you don't want to degrade? No? How about if the FOX protein is involved in the cancer metabolism, we want to inhibit it. If the FOX variant, it's called a tumor suppressor. So in other words, some FOX proteins are tumor suppressors. In other words, a tumor suppressor is a molecule that whenever it's present, it inhibits tumor growth. So we don't want to inhibit tumor suppressors. Regrettably for us, FOXO3A is one of those tumor suppressors proteins. So we see some degradation on it. So we need to do some more structure-activity relationship to find a Protac that only inhibits FOXM1, FOXA1, FOXK2, but not FOX3A. So we start seeing, and then I'm sure they're going to comments I'm going to get from reviewers in the grant here. You don't have selectivity. Come back to us when you have a better drug, right? So with that, I'm just going to end because I only have I'm over 2-3 minutes. Just thank you to all the people who are involved in this. My two students in postdoc, Antonio Vega and Erika Loredo. This is Dr. Karp, who is helping me with all the ovarian cancer work that you saw, and his student Mackenzie. And I'm open to questions for you. Thank you very much. Appreciate it.
Muchas gracias. Muchas gracias, Carlos. Thank you so much. We have time for a couple of questions.
What are the main disadvantages of the use of Protac?
Main disadvantages of use of Protacs. High molecular weight. The pharmacokinetic, I didn't have time to show you, but some of these Protacs have low half-life, anywhere from 7 minutes to the most stable Protac, it's about 3.5 hours. But for most of them are short, especially when we test it with liver microsomes. when we do a proper pharmacokinetic testing. Some of them, because they're big. So remember that we have 2 drugs and a linker. So whatever pharmacokinetic you have, multiplied by 3. So because 3 drugs are being degraded, are metabolized at the same time, right? The hook effect is something also, another disadvantage. As when we have to find the right concentration when we're trying to treat the disease, we have to be able to find the right concentration. Otherwise, the trimer starts dissociating and is no longer effective. Those are probably the 2 main disadvantages.
Thank you.
I was just going to ask a related question. So I think a potential disadvantage is cell permeability. They have to get into the cell.
Yes.
So I think that has to be difficult with the large molecules. Related to that, this hook effect, couldn't it also be an aggregation effect? So that, certain as you build a concentration up, like the Protac is aggregating, so then there's less free Protac, or it could be just aggregating with other components of the cell.
Absolutely, yes. Absolutely. Cell permeability is one, another disadvantage because, but surprisingly, despite being the molecular weight of Protac, the average molecular weight's around 700th, 800th. So they're big molecules. But despite that, they're effective. Even though they some degree of cell permeability, even if a little bit crosses the cell membrane, because of that cell, that cycle that recycles the Protac once and again and again. And in this particular case, it's even more notorious because FOXM1 regulates its own expression, right? So whenever you have that case, when a protein regulates its own expression, when you start degrading it or you start inhibiting it, it no longer replicates or it no longer expresses itself. So the concentration goes down, even if it's just a little bit of cell permeable Protac. Yes. There are some examples. They're trying to develop oral Protacs. It's not easy. Yes, it looks like most Protacs are gonna have to be injected, but the main challenge here is Arvinas, amongst other companies, are trying to develop orally available Protacs. But they're gonna get there, yeah, eventually.
Carlos, thank you for a really, really nice talk and really nice work that you guys are doing. When you talk about the advantages of Protacs, I think something that is interesting to share is that when you have a protein that has 2 different functions, for example, has an enzymatic function and a scaffolding function, and you don't know which one you want to inhibit, a Protac is perhaps the best tool you can get because it's going to take everything down and then you're going to be able to really test what happens when you completely remove that protein. So that's, and then a question for you. You did not show us the source of the binder or your ligand for FOXM1. Was that kind of an in-house molecule or
yes.
is that coming? And
yes.
do you have work trying to find a more higher affinity ligand for those for FOXM1?
Exactly. Yes, that's what we're trying. We're just getting started with this. We've out of the 96 Protacs that I show you, we've tested about 9 or 10. So we still have a long road, but we're looking for that gives us the ideal profile, right? Yes.
So let's thank again, Dr. Carlos.
Thank you. Muchas gracias. Entonces me desconecto. Listo. Muchísimas gracias, José Luis. Muchísimas gracias. Tengo unas preguntitas. Muchas gracias. Mientras te puedo ir presentando.
Sí, si quieres unos minutos nomás
Claro.
para conectar y listo. Y es para conectar al Ok. Y creo que uno de estos sirve para avanzar, ¿verdad? Sí, a ver si no. Bueno, vamos a ver, si no.
Our next speaker is Agustín Casimiro Garcia. Very briefly, Agustín Casimiro did his undergraduate studies here at UNAM, QFB, under the supervision of Dr. Rafael Castillo. Then he did his PhD in medicinal chemistry at Purdue University. And then he moved to New York to do a postdoctoral stay at the Rensselaer Polytechnic Institute. So he has a very large professional experience. Actually, he started as a research assistant here at UNAM. Then he moved to Pfizer. He worked in medicinal chemistry. That was in the year 2000, where he's actually still working in a medicinal design area in Cambridge, Massachusetts. Dr. Casimiro is affiliated with American Chemical Society in the Organic and Medicinal Chemistry divisions. It's peer reviewer of several very important journals in the area, and he has received several honors and awards. Just a few of them was actually Gabino Barreda Medalla Gabino Barreda in 1994. Then he also got a prize at Purdue University, the John Division Award in Medicinal Chemistry. Then another award at Purdue University, the Jenkins Nobel Award for Excellence Research. And in Pfizer, he got the Pfizer's Neurotonin Team Award. He has 35 publications, 15 patent applications, 8 presentations, and has participated in 13 meetings. Thank you so much, Agustin, for being here.
Buenos días a todos. Good morning, everyone. So again, para poder que sea esta plática más accesible para el profesor Jorgensen, vamos a ponerla en inglés, así que voy a empezar a hablar en inglés. Good morning, everyone, and thank you first for the very kind invitation to participate in this symposium. Really, for me, it's an honor to participate in this symposium, especially because it is dedicated to a very special friend, a very special person, Dr. Rafael Castillo Bocanegra. I'll come back to that at the end of my presentation, but let's first start with about what this is talk about. So, as the title of this presentation is, this is a 3 drug discovery stories. So, I'm going to tell you first a story related to fibrosis, where we work to identify a hit compound, and then we advance to an in vivo tool. Then, in the second project that I'm going to be discussing, this is a project related to a genetic disease called sickle cell disease. In this case, I'm going to be telling you about the identification of a hit compound and how we use that hit to advance it to a clinical candidate. And finally, in the third project, I'm going to be telling you about JAK3. And this story, some of you may actually be familiar because I had the chance to share kind of an early story about this project in 2016 at this symposium. I shared some of the advances we have accomplished at that point. Later on, in 2021, I gave another advance when there was an opportunity to share this at the Facultad de Química. Today, I thought that this was a very interesting story to share also because there's a very, very interesting development with this story. So now you would ask, why include 3 stories for this presentation? Well, there's a reason for this. And the reason is that one of the most important steps in the life of a drug discovery project is the identification of new chemical matter, of the series that you're going to use to really run that project. And for this, there's different methods, and this is kind of an interesting review from 2019 of the different methods that you can use for identification of chemical matter. So today, I pick up 3 different projects that sit in different places in this kind of map of sources of chemical matter. The first project, PMT, uses a phenotypic screening. The second one, HPS, uses an vitro screening that we have heard already a lot today. So I'm going to show you another application of this methodology. And finally, the third project, see a big difference where it sits, is where you have already a quality lead, and then you advance that quality lead to get you to a candidate. So I hope I don't have to ask people to stop watching their phones. I hope we get your attention. So hopefully I don't have to do that. But let's start with the first project. So again, first project is called PMT, and it's related to fibrosis. So what is fibrosis? Fibrosis is a disease that impacts many different tissues. You can find fibrosis in the liver, you can find cardiac fibrosis, kidney fibrosis, etc. So it's a very serious problem. It usually starts with an injury in the tissue. Then there is a secretion of a lot of the extracellular matrix components, and this leads to a progressive changing of the organ, all the way to completely destroying the organ architecture. And usually at the end is loss of function. For example, in the case of liver fibrosis, that develops all the way to getting, for example, cirrhosis. So it's a serious problem. And another important problem is that there's very few drugs for treating fibrosis. In fact, for one form of fibrosis called idiopathic pulmonary fibrosis, there are only 2 drugs, and none of them actually cure fibrosis. They're only kind of to tune down the symptoms of the disease. In the case of liver fibrosis, there was not a drug until I would say probably the last few months with all the GLP-1 agonists. They start to show some effect on liver fibrosis. But really, if you want to find an area of high unmet medical need, this is fibrosis. But it's very challenging. Many companies, many groups have tried to get something for fibrosis, and they have failed. So it's a very, very difficult area. One of the steps that happen during this process is the transformation of one cell type called fibroblasts, that they're converted into what we call an activated fibroblast, and they start secreting those molecules that are involved in the extracellular matrix. This is collagen and another protein called alpha SMA, or alpha smooth muscle actin. So today, I'm going to be talking about liver fibrosis and how we use this collagen to try to find an opportunity to modulate and to work around fibrosis. So in liver fibrosis, there's one particular cell type that is very important, and this is called the hepatic stellate cell. This has been known to have an important role in the establishment and in the process of fibrosis. If you look at this figure on the left side of the slide, you will kind of understand what happens with these cells. You have these quiescent hepatic stellate cells, and in the presence of pathological and relevant stimulants of fibrosis, one of them is called TGF-beta. In the presence of these substances that happen during the fibrosis event, these are transformed into these cells that you see in the middle, myofibroblasts. These are activated cells that are characterized by increasing in the secretion of these molecules that I mentioned before, alpha SMA, collagen, also contractility, proliferation. So we identify this step for potential intervention, and then we ask the question, okay, so what if we target this event with a phenotypic screen to try to find molecules that will have an opportunity and a potential therapeutic agents for fibrosis? Again, going back to the figure, it's kind of the same thing. We're going to run a screen where we're going to start with the cells that I mentioned, hepatic stellate cells. We're going to use relevant stimulation, either TGF-beta or thrombin, and then we're going to read the changes in the cells using an assay. We use an assay that is called a high content screen, and we use immunofluorescence for the detection of those 2 molecules, alpha SMA and collagen. So you see kind of the colors, how you actually visually detect those 2 molecules. We ended up with one more readout, which was cell count as a measure of toxicity. And we actually collaborated for this work with a company in Germany called Evotec. Fantastic partners, really great experience working with them. So this was our screen and our screening funnel. All the different assets that we start to use, that we use for really going from about 20,000 compounds all the way going to a number of hits that was more manageable, as we have seen already from previous presentations. I'm not going to tell you about all the different steps. I'm just going to comment that we use 2 different stimulants, TGF-beta and thrombin, because we thought that will give us the opportunity to explore a broader biology and looking for potentially better opportunities to interfere with fibrosis. We also use something that is called transcriptomics, and for this we took the cells, treated the cells with the compounds, and then look at the gene transcription changes with each compound, and then look at that change. For that also, we have reference compounds with no mechanism fibrosis, and then we were able to compare the changes at the gene level for each compound compared to the reference, and then in this way it say, okay, if a compound shows a mechanism that seems to be something that is already known, we say we're not interested in that mechanism. So we were able to use as a filter. To make this story short, we ended up with a series that we call the sulfonamide series. And this is a hit that we find as a hit. I'm going to share kind of the characteristics of this hit that got our attention. First of all, we measured collagen, as I mentioned, and this compound actually show a selective effect with collagen, both with thrombin and TGF-beta, and an effect somewhere between the 1 micromolar to about 2 micromolar. Interestingly for us, this compound was selective versus alpha SMA, no effect on alpha SMA. Okay, we say, okay, something different. Now, this compound came from one of the collections that we have in-house that we call the drug set. This is a collection of compounds that are drugs that are currently in use, and also compounds that were advanced to advanced clinical studies. So because of this, we thought, this compound should have good properties. So, represents kind of nice chemical matter. And in fact, this is a known endothelin receptor antagonist. And in fact, it's quite potent, with a potency about 1 nanomolar for this particular target. However, we did a lot of work to really ask the question whether endothelin receptor activity is related to this PMT effect. Basically, and I probably skipped that part, but it's the transformation of that initial cell that I mentioned. So we asked that question, and our work showed that endothelin receptor activity is not involved. So this is not driving the phenotype that we're reading in this particular assay. A couple more characteristics that got our attention. I mentioned that we use that transcriptomics screen to really compare versus known compounds with known fibrotic effects. And this compound showed no effect at the transcriptomics level. Pretty much was very similar to DMSO. So that was also something kind of intriguing to us, because we had also plenty of other compounds that have a very strong at the gene expression level, but many of them were toxic. So this was not very attractive to us. So that was kind of another characteristic that we consider interesting. Then we started to think about, let's think about the collagen biosynthesis pathway. What are the potential steps where this compound could be interfering? This is kind of a nice reference, very nice illustration of all the biosynthesis steps for collagen. During that time, we also used another technology called proteomics, that basically you can look at the compound, at the changes at the protein level when you treat a compound, when you take the cells, treat it with the compound, and look at all those proteins, all the protein map, and see which ones are changing. One thing that got our attention is that this compound appeared to change the hydroxylation of collagen 1A1. This kind of taking all this data together suggested that perhaps we were impacting collagen biosynthesis at the level of post-translational modification. So with all this information, we say, this is an interesting compound. We should continue in advancing to do additional work. During this time, we were also interested to say, okay, we have a phenotype that we're reading in our assay. It's all in vitro assay. So what happens when we take a molecule that comes from this screen and put it in more complex models? Is the effect going to be sustained, or is it going to be lost? But for that, we needed a series that we can advance quickly, that had the right properties. And we said, this is a compound. It comes from another drug. Chemistry should be kind of well established, so we can advance it quickly. And therefore, we advanced with this compound. So our goal was to find a tool And for this, we look at the properties of this compound. Potency, as I mentioned, around a micromolar. Because we were interested in finding a tool for a mouse in vivo model, we look at the metabolism of this compound in mouse. So we use mouse hepatocytes or mouse liver microsomes. This is high clearance. So this was not suitable as a compound to advance for an in vivo study. So our job was finding a tool that in a molecule that we have reasonable potency and better, preferably very low clearance, so that we can actually put it in a PK study. So we prepare a little bit over 500 compounds for this series. Certainly, I'm not going to share all the compounds. I'm going to share just a few of the things that we did around this compound and how we started to learn about the phenotypic activity. And keep this in mind. This is all phenotypic activity. This is not a target base, okay? We're very used to working with a target, with a known target, but we are working based on a phenotype, a change that we're looking in the cell assay. So, but we look at the changes on the sulfonamide and the dimethyl isoxazole fragment, we saw that this actually killed the activity. We know we couldn't change that fragment of the molecule. Then we look at the bottom portion, the dimethyl isoxazole replacement, and we started to see some changes. We started to see some gaining potency, so which was good, and particularly the compound 2, which is on the far bottom right corner of the slide, we started to see better potency and lower clearance. So we were thinking, hey, this is going in the right direction. Then we look at changes in that phenyl ring that is next to the sulfonamide, and we saw further improvements in potency, all the way getting down to 2 double-digit nanomolar, which nice. However, the clearance was still high, so it's like, okay, still not there. So then we took one more drastic change in the molecule. And think about when you think about molecules, sometimes you have to make drastic changes. So and you need to keep that in mind. There's many tactics, right? Sometimes you make a ring, sometimes you break a ring, sometimes you remove rings in order to find the right compound. Well, for us, a truncation strategy worked better because at the end, compound 3 listed there is a compound that ended up being kind of the most appropriate compound to move forward. A compound that retained acceptable potency, 230 nanomolar, with, especially with the mouse hepatocyte and mouse liver microsomes very low, basically below the level of detection of our assays. So this compound was cleared very, very, very slowly. So it was kind of the right compound. Now, when you work again on phenotypic screening, you don't know the target. We did some work to try to find the target, we couldn't. But then one of the questions that emerged at this point is, are these compounds like compound 2 and compound 3 really related? And can they interact with whatever target we interact in the same fashion? Of course, you don't have a crystal structure, you don't have anything to guide you, right? So you have to trust or use other approaches to try to at least in some way answer that question. The way we did it is compare energy minimized structure of compound 3, which was the first approach. We used, did a QM calculation to get at the structure. It's shown on the figure on the right. That bold orange structure is the energy minimized structure of compound 3. Then we compare it. In our data set, we have lots of crystal structures of molecules, and we compared that with the structures of the biaryl or biphenyl sulfonamides. And for the most part, they look similar. It seems at least they were occupying similar space. Went back and compared now to just monoaryl compounds. And again, we saw, this looks similar. So it seems that at the end, our conclusion was that at least compound 2 and compound 3 can occupy at least a similar 3D space. It was kind of our best approach to try to answer that question. Not perfect, but again, that was kind of the only approach. Okay, a little bit of synthesis. I think it's good to think about that this happens only when you prepare compounds. It doesn't happen another way, so we still have to make the compounds, put it in the vial, and then test those compounds. So as I mentioned, chemistry for these compounds was easy because of the known, it's coming from something that is known. So on the left, you see the use of parallel medicinal chemistry to quickly advance to the, to make compounds. So we use different types of chemistry to make compounds. We were able to test different design hypotheses in this way. On the right, you see then more single target synthesis. The chemistry was easy, right? So a couple of steps and we were able to get the compound. Compound 2, for example, we start with sulfonamide formation. then Suzuki coupling to make the biaryl system, and then basically protecting group removal. Very easy. Compound 3, which is a compound that we move for in vivo study, even easier, basically 3 steps, and we were able to make lots of compounds. At the end, we ended up making over 10 grams of compound to basically move to the in vivo studies. So this is just a summary of compound 1 and compound 3. So going from the heat to the tool. We saw an improvement in potency, that's the first row, going from 1 micromolar to about 230 nanomolar. We retain some reasonable properties. We use a panel of assays that is called CEREC. This is a collection of enzymes, receptors, ion channels, and this is a good way to understand if the compound is selective or is a very promiscuous compound. You see that compound 3 was very selective. We didn't see much activity. Also, think about this series comes from endothelin receptor antagonist. It was important to determine if compound 3 still retains some effect, and we demonstrate that basically this compound also was inactive for endothelin receptor activity. Tested in mouse PK studies, data was adequate to continue moving forward, and then we use human data to do a dose projection in mouse, and I'll comment that in a minute. But basically, we projected the dose that was needed to achieve either 50% or 80% inhibition of collagen in the animal study. And someone is gonna say, what? What did you do? Why is it you use human data to project a mouse, an in vivo study in mouse? Well, unfortunately, that's one of the risks that we have to take because we couldn't get a PMT assay in mouse. So we have to take that chance and say, okay, we're gonna go forward, we're gonna take it, and there is a risk that even the biology that we're potentially targeting with this compound doesn't happen in mouse, right? So that's a risk. For that, as we move for an in vivo study in mouse, we also advance this compound to another ex vivo assay, but using human tissue. So it was a way for us to balance that risk and say, okay, we're gonna try to see what happened between the 2 assays. Okay, this is in vivo model that we use. It's called the carbon tetrachloride. We chemists know very well. It's toxic to the liver, right? So it's very well known. So that's very clear why we use that model. It's actually a very well established model of liver fibrosis. I don't want to say it's a good model, but it's a model that is used. We use it in 2 different modes. The first one, we call it therapeutic, because in this case, we allow the disease first to establish for 2 weeks after you give carbon tetrachloride, and after 2 weeks, you give the compound, and then look at the effect of the compound for 2 weeks. On the second, it's a prophylactic approach, where you give compound and carbon tetrachloride at the same time. You let it go for 2 weeks, and find out what happens. We measure, we run all the PK studies to understand that we're reaching the concentrations of the compound that we're hoping to accomplish. And you see with the 20 milligram per kilogram BID dose, we were able to get about 90% inhibition. So we felt that we have basically got as much inhibition of collagen with that compound at that particular dose. And unfortunately, this story doesn't have a happy ending because at the end, what I'm going to show you is that by 2 different methods, both histopathology analysis and proteomic analysis, this compound did not work. So we measure in the histopathology, we measured the amount of collagen that was being inhibited. Basically, we didn't see any change really. And in terms of the proteomic assessment, this also did not show any change. So you see, it's not all success. There's many failures in the pharmaceutical industry. And this is one example where we spent a lot of effort and didn't take us to any success. Well, you can ask, okay, this again is a mouse model. Okay, maybe the biology didn't hold between mouse and human. Well, for that, we run that other model, an ex vivo model. It's called precision cut liver slices. They use liver slices from human tissue to run that at the screen. And unfortunately, in that screen also, we did not see any significant change in collagen by this compound. So unfortunately, again, this story doesn't have a happy ending, and this led us to stop the work on this series, and then basically later on to stop the work on this phenotypic screen. So again, one of the challenges of the industry, there's many failures, believe me. You probably also see this very often because we're used to that, and it happens very often. It's probably more failures than actually success. It's very challenging. Okay, let's move to the second project. I hope everyone again is awake and I still have your attention. So the second project, you will see some very interesting things in this project that perhaps you may never see again in any medical project, and I will tell you why in a moment. So first of all, a little bit of the disease. This disease is called sickle cell disease. En español it's called la enfermedad de las células falciformes. Kind of a strange name, but you know, that's the name. Why the reason for the name? What happens in this disease, there's some mutation in hemoglobin, and this mutation in hemoglobin leads to the red blood cell to change the shape. And you see kind of on the top of the figure that how the red blood cell is changing the shape to what is a sickle. And look at the sickle kind of on the top far right of the slide. That's what is a sickle, and that's why it's called the sickle cell disease. Now, what happens with this mutation in hemoglobin? Well, this leads to different problems in the vascular system, vasoconstriction, hemolysis, etc. So at the end, there's significant organ damage and usually mortality. I think what is more sad is people that have this disease have approximately a life expectancy 20 years fewer than you know, a normal person. So it's a serious problem. Now, to understand better the disease and what we try to do for this particular disease, we need to understand a bit better about hemoglobin. Hemoglobin has two chains. Let me actually take some water before I continue. Give me a moment. Hemoglobin has two chains, 2 alpha and 2 beta units. And remember that hemoglobin circles between oxyhemoglobin and deoxyhemoglobin. So in the case of sickle hemoglobin, there's a mutation in the beta unit. And this mutation leads to creating lipophilic surfaces or lipophilic pockets in that beta unit. This then leads to basically start a process where there's a formation of a dimer, and then a nucleus of a dimer, and then overall, you know, a formation of a polymer, which is shown here on the bottom left of the slide. Now, this nucleation only happens in the deoxy state. So this disease, there's a high concentration dependent effect. So even a small decrease of deoxyhemoglobin, sickle hemoglobin, then this can actually lead to a kind of a nice delay of the disease and a significant improvement in the disease. So at the end, our goal was to find a compound that can stabilize the oxygenated version of hemoglobin, and in this way delete the process of this disease. Now, this is, again, some of the very particular characteristics of this project that, is very important. I think keep those in mind because, again, I don't think you're going to see situations like this very often. Before, let me take you through what is a typical standard small molecule project. Usually the characteristics. Usually, the drug is in large excess of the target because the target usually is a very small concentration, usually less than nanomolar. Also, when you're trying to optimize a dose for that compound, you usually use either potency or clearance. And finally, you typically go for occupying the target as much as possible. Sometimes we go for IC99, sometimes IC95. So you want to inhibit as much of that target as possible to get you your therapeutic effect. Well, what happens in this project, sickle hemoglobin? Well, first of all, the target is in large excess of the drug. Why? Because hemoglobin is found between 1.8 to 2.5 millimolar in the red blood cell. So just think about the amount of the target that you have. Third, actually, occupancy is going to drive the dose. It's not going to be the potency and the clearance. It's all about occupancy. You want to occupy, you target as much as possible. And third, you only need a very small amount of target coverage to drive this effect. 10 to 30% is enough. We learned that through either some initial studies that were done in this target. So again, very particular effect, very particular characteristics of this project. And the next one is actually shown in the next slide. First of all, for identification of chemical matter for this target, we utilize virtual screening, and I think we have seen already lots of, actually heard very good talks about virtual screening, so very good posters. So we utilize this technique to actually find this chemical matter. We screened 2.5 millions of compounds using virtual screen. From here, we ended up with 35,000 compounds with good docking scores and actually lacking undesirable features. And then we actually run the actual screen for activity utilizing a methodology called affinity selection mass spectrometry. Out of this, we identified compound 1 as one of the hits. We had more hits, but this was one of the hits that was interesting. And compound 1, you will see there's a chiral center. So we quickly did separation of these two compounds. And actually, we realized that the eutomer, which is the active enantiomer, is DS with a binding affinity of about 3.6 micromolar. We utilized a technology called surface plasmon resonance for measurement of the binding affinity, 3.6 micromolar. Okay, as we continue the profiling of this compound, we utilize different biophysical methods to characterize. One thing that quickly was observed is that there were 2 units or 2 molecules of this compound binding to hemoglobin. And later on, we were able to confirm by X-ray crystallography that in fact, there are 2 molecules of this compound binding to hemoglobin, one to the unit alpha 1, one to the unit alpha 2. So this is called ditopic binding. So it is very, very uncommon to have molecules that act by this mode. So if you happen to find another one, just make a list, because again, there's very few that actually happen, very few. And in fact, I haven't ever seen another type of binding like this, like only what we observed in this project. Look also at the very interesting pi-pi stacking that is observed in the crystal structure that is formed between the aminopyridine pyrrosol motif of each one of these molecules that you see, that really, really nice stacking that you see in this case. And this basically places those molecules for kind of really nice interaction with the 2 alpha units. Okay, so we have a hit. This is kind of a little bit more of the characteristics of the hit. Binding affinity, 3.6 micromolar. We use another assay that is called the R2T, where we see the change between the oxygenated state of hemoglobin to the deoxygenated state. The goal is to delay that, right? So we want to keep that hemoglobin oxygenated for as long as possible. This compound shown effect about 21 micromolar. Okay, so we started the optimization process. We made modifications on the top, modifications on the bottom. It seems that we were not making much progress. Then we made an interesting observation. And this is something, keep in mind those things, because you want to look at that interaction of your ligand with the crystal structure. Sometimes you find something interesting, something that you can use to, make a better, changing the direction of your series. In this case, what we observe, if you look at the structure, there's on the bottom of that surface map, there's this orangish area that represents a lipophilic area in that protein. And what we hypothesize is that, okay, if we cyclize that aminopyridine to form another ring, it is possible that we're going to present more lipophilic surface and therefore have a better interaction with that surface in the protein. And also, we could even engage in better pi-pi stacking, because a few 6-member ring actually can lead you to better pi-pi stacking. So the proposal was go from that aminopyridine motif, build a ring, and get into that aminoquinoline series. So that actually worked well for us. Our first molecule we prepared, we started to see even a small change in the affinity. But we recognized that we were losing the hydrogen bond donor and acceptor moiety in the pyrrosol. So we hypothesized that we needed to recover that in some way. So we placed either a nitrile or an amide. And as you see, now we start to see kind of the change in the direction. So compound 2 now we're showing a 10-fold gain in potency. In order to check that we haven't changed the binding mode, we got another crystal structure, and it confirmed. Still ditopic binding, still that nice pi-pi stacking between the 2 molecules. Okay, we continue the optimization process. We start with this, looking at this region of the molecules where that fluorophenyl ring is located. So we started to do some modifications there, and we find this compound 3, that started to continue some improvement in the binding affinity. But we started to pick some effects that we were not interested in keeping. This is effect on ion channels, which was also demonstrated in the hair activity. So it's like, this is not the direction. So our hypothesis at this point is that we needed to play more with the polarity of the compound. The polarity that we have included in the shape of that moiety on the right of the molecule, we thought, it's like, what if we move that polarity to the other side of the structure? Let's try that approach. And actually, again, this worked, again, very effectively, where we actually introduce a carboxylic acid in that region of the molecule, and look at the very significant change in affinity for that compound. Now we're getting to basically single-digit nanomolar affinity, Kd, for that particular interaction. What was more interesting is that acid led us to create new interaction or engage with new interactions with hemoglobin. You see that now that carboxylic acid interacts with arginine 141 and asparagine 78. Great, we were making very good progress. We have found compounds that are more potent. We continue to work with acids. For example, acid 5 is another molecule that showed very good, very good potency. But during some additional studies with these acids, we started to see formation of metabolites that was a concern. So we decided to take one step back and go back to neutral space. Think about when you're working with molecules, think about you have your neutral space, you have your space where are acids, you have a space where you can work with bases. So in this case, we decided to go back to a neutral space. After doing some new designs, this pyridone compound 6 is a compound that became the winner of this story. This compound retained good binding affinity, 34 nanomolar, and we confirmed that pyridone moiety still engages the same amino acids that I showed you before, similar to the carboxylic acid. So this was, again, the winner of this story. The compound was advanced for additional profiling. This is just a little bit of the data. In terms of the KD, I mentioned 34 nanomolar. We have also made significant improvements in that R2T assay with 4.8 micromolar. The initial hit was about 20 micromolar, so we moved that number significantly. But think about that this story, as I mentioned at the beginning, was about occupancy, right? Remember, for a normal project, it's about for this project, it's about occupancy of hemoglobin. We want to occupy hemoglobin as much as possible, and we want to be in that red blood cell because that's where is our target. So the number that you see here for the red blood binding, this compound has a fraction bound very small. Compared to that, to how much of that compound is bound, you ended up with about 99.95% of that compound is bound to the red blood cell. So exactly basically the profile that we were aiming for. Very quickly, a few review of the synthesis of this compound, or at least a few of the steps on the synthesis of this compound. We start with a Mitsunobu reaction to form the ether bond. The second step is utilizing the activated quinolone as the N oxide for introduction of the 2 amino group. Then we do a bromide to methoxy transformation, and finally, using just basically demethylation of that methoxy group, we end up with the pyridone, which again, remember, is in equilibrium with the hydroxypyridine. For evaluation of this compound, we utilize a model that is called the Towns sickle cell disease mouse model. Think about a humanized version of the disease in mouse. The genes, the human genes, are knocking into mouse and creates this model that then you can use to test these compounds. The first thing we did, which is shown on the left of the slide, is about the pharmacokinetics of this property. We wanted to make sure that we have enough concentration of that compound. So this was followed for several days, and we demonstrated this compound achieved the right concentrations. And we got up to 60% coverage. Remember, we were targeting even a small coverage, so 60% was enough for us. The panel on the right, or the figure on the right, shows you the effect of the compound on stabilizing the oxyhemoglobin. Remember, that was our target. It's stabilizing the oxygenated form of hemoglobin, because that then will lead to a delay in the disease. The graph on the right shows you the effect on P50 which is the oxygen pressure at which 50% of hemoglobin in a sample is oxygenated. So if an agent is going to stabilize the oxy version of sickle hemoglobin, this is going to lower that P50 value. So if you see on the graph, the line in blue is the effect with vehicle. The line in green is the effect with compound. So you see a very reasonable decrease in that effect. And at the end, we calculated that to be about a 50% effect on reduction in that P50. So again, that was looking very promising for us. And then in the same model, but now looking at really the number of sickle cells, using again high content screening, we're able to compare the effect of the vehicle versus compound 6. And really, as you measure that effect that is shown on the right, this is about almost a 40% reduction in sickle cells with this compound. So this, along with a lot more data, was used to support progression of this compound to the clinic, to a phase 1 study. And I'll come back later to tell you, what happens with this compound. Okay, let's move to the last story of this project. And again, some of you may recognize pieces of this story, but I think, again, there's some interesting progress with this project. So let's first a quick review on JAK3. So JAK3 is a member of the Janus kinases. There's actually 4 different JAKs, JAK1, 2, JAK3, and TYK2. They are involved in cytokine signaling. What happens is that cytokines that are shown at the top of this figure binds the receptor. Then the JAKs, which work in dimers, are phosphorylated. After phosphorylation, they interact with another set of molecules called the STATs. These STATs also get phosphorylated, get to the nucleus, and have effects on gene transcription, or basically transcription regulation. Okay, so we became interested in the pathway signal by the JAK1, JAK3 heterodimer, because our hypothesis, based on the function of this heterodimer, this could have potential indication in different autoimmune disorders. And I have here 2 examples. One is alopecia areata, the other one is vitiligo. These are 2 autoimmune disorders, and I'll come back towards the end of the talk about alopecia areata. So this was our hypothesis. We need to inhibit this pathway. So how would you inhibit this pathway? Okay, you can think about doing the inhibition of JAK1, find a selective JAK1 inhibitor. But what happens with JAK1? If you look at the graph, JAK1 associates with other JAKs. So if you inhibit JAK1, you're going to be inhibiting several of the other pathways, and therefore you're not going to get selectivity. You're going to have effect on other pathways. This could lead you to some type of side effects, as we know, happen very often with drugs. So okay, so going back, so JAK3, we need to find a selective JAK3 inhibitor for inhibition of this pathway. Okay, that became our goal. During the time of starting this project, there was actually work that suggested that the selective inhibition of JAK3 would not be sufficient. And in fact, our competitors from Novartis published a paper where they say, no, forget about JAK3. JAK1 is the only one that's going to drive this efficacy. So we decided to take the chances. Pfizer has been a pioneer in the JAK area. So we decided, I think we have enough knowledge that this should work, and we decided to go. So the task was finding a JAK3 selective inhibitor. Okay, so we target the ATP binding site of JAK3, and for this, it's important to remember that this part of JAK3 of the JAKs actually has extreme similarity. They're very basic, pretty much identical. Look at the different amino acids at different positions in this ATP region. Pretty much they're the same. Fortunately, if you look at that area that is highlighted, in JAK3 there's a cysteine 909. In the other JAKs there's a serine. This was kind of the area where we say, okay, there's a difference, and then we can target this cysteine with covalent inhibition. That was basically the approach that we follow. Before I go and tell you about that, I just want to share a little bit about this other compound called tofacitinib. Tofacitinib is a JAK inhibitor, also discovered in Pfizer, is currently used in the clinic. It's an approved medicine for different autoimmune disorders. initially reported as a JAK3 inhibitor. Later on, it was really confirmed to be primarily a JAK1 inhibitor with effect across other JAKs. So at this point, you can say it is a pan-JAK inhibitor. This was the origin or the lead matter that then we used for taking this series for covalent inhibition. So in terms of covalent inhibition, what we're doing, so we have a cysteine 909. It has a reactive file that can react with electrophilic molecules and then engage in covalent interaction. So if you look at the two panels on the right of the slide, we utilize two different types of electrophilic molecules. One containing alpha-beta unsaturated amides using Michael addition, and we end up with these type of molecules are going to lead you to covalent irreversible inhibitors, and keep that in mind, irreversible, because in the case of, in the second case with nitriles, cysteine also react with nitriles, but these lead you to covalent reversible inhibitors. So there's an important difference between the two. As you look also at cysteine 909, it was also important to understand that there's 10 other kinases that have a cysteine at exactly the same position. This is the list. Many of these kinases are part of the TEC family, and I'll tell you later how this, came back to us later as we continue. Now, part of understanding and the challenges of going in for this particular target was related to safety. One, I mentioned there's 10 other kinases where this compound could actually interact, could lead us to some side effects. This compound also can react with other cysteines in other proteins, right? I mean, there are cysteines pretty much in all proteins. So this was a risk in terms of potential protein modifications. Another characteristic that was important when we run this project is something we call biochemical inefficiency. And this comes from the fact that JAK3 has very high affinity for ATP. And therefore, when you run a biochemical assay, the concentration of ATP that you use is very important. If you run the assay at low ATP concentration, there's gonna be a significant shift when we take that compound and test it in a cellular assay. And we showed here in this graph the assay on the x-axis is run at 4 micromolar of ATP. The assay on the y-axis is run at 1 millimolar of ATP, a much higher concentration, closer to the cellular concentration of ATP. And if you compare the data from the two assays, there's about 100-fold shift between the two assays. So you could be running compounds at the low ATP assay, and you run to your cell assay and say, okay, these compounds are not active or are much weaker, 100-fold weaker in your cell assay. So it was very important to run the assays at the appropriate ATP concentration. And finally, it's very important to remember or think about the resynthesis rate of JAK3. Why? Typically, for a covalent inhibitor, there's an advantage that with a protein that has a long resynthesis rate, you're going to use your covalent inhibitor, it's going to bind your protein, and if the compound lives for, if the resynthesis rate of that protein is 24 hours, well, for 24 hours, that protein is not going to function anymore, right? So it's going to have a prolonged PD effect or pharmacodynamic effect. Well, unfortunately for us in JAK3, this doesn't happen because it has a resynthesis rate of about 3 to 4 hours. So we're losing that. So at the end, in order to get a good low dose, we go back to the same concepts that I mentioned in the talk before. Potency and low clearance. So we needed to find compounds with the right potency and the right clearance. Okay, as I mentioned, our inspiration or lead for this project was tofacitinib. We had access to crystal structure, so we were able to see how this compound bind. One thing that was observed, if you do a rotation between the nitrogen and the pyrrolopyrimidine ring and rotate that molecule, you will see that the amide fragment of this molecule is going to come closer to cysteine 909, and therefore it's going to be kind of presenting a potential electrophile in the ideal position. Well, this is exactly what we find out when we prepare a molecule like that. We got to target cysteine 909, perfect. We also, took some time to understand that, the substitution both on the piperidine and on that nitrogen of that nitrogen of the piperidine was important. And for example, we run calculations and showed that the hydrogen at that position, R1 in this particular scheme, actually promoted a conformation that was more favorable for interaction with cysteine 909. Experimentally, we demonstrated that. If you look at the first two compounds in the table, the first one retains a methyl group on the piperidine, a methyl group on the nitrogen. Pretty much no much activity on JAK3. Second compound, we just keep the methyl group on the nitrogen, again, no activity. Fortunately, third compound, no methyl groups on either one of those positions. We got to basically pretty much very good potency for JAK3 and no activity for JAK1, so very high selectivity. Interestingly, this effect, could also be retained using a conformationally retained analog that is shown here. This compound, we got also crystal structure for this compound. We showed that this conformational restriction effect also led to a compound that showed pretty much the same effect as the compound that I mentioned earlier. Now, at this point, I think we had a good understanding of how to optimize for potency, for selectivity based on these compounds, but then we were, I would say, struggling or really having to learn about more the metabolism of these compounds, the clearance of these compounds. And for this, we use these two compounds. Let me check the time. We utilize these two compounds. One has a microacetyl compound A. Compound B is just an acetamide analog. We use and run these compounds through multiple assays to understand the clearance pathways of these compounds. We got to understanding that there's two components: the typical P450, or usually driven by in hepatocytes, but also there was the extrahepatic clearance. And this usually is driven by a group of enzymes called glutathione S transferases that conjugate glutathione with an electrophilic compound. We have to develop assays to really being able to modulate this activity. And for this, we utilize blood stability. That was kind of enough to tell us about the component of that extrahepatic clearance. Using both hepatocyte data and blood stability, we were able to beat correlations that are shown on the top right between calculated clearance versus actually experimental clearance. And you see that the correlation was good. So, okay, we had understanding. Now, what we didn't know is how to modulate the extrahepatic clearance, the GST modulated activity. And for this, we were working with a second series called the cyanamide series. If you see these two compounds on the top, cyanamide A and cyanamide B, the only difference is a methyl group that is next to the nitrogen, near to the cyanamide, on the top of the molecules. It's highlighted in blue, it's very light, but it's there. So there's a methyl group. And after tasting these compounds, and again, in a panel of assays, what we observed is that small transformation led us to better stability in blood, and also to a reduction of reactivity versus glutathione. And this was kind of the key concept. A steric hindrance near the electrophilic group then was sufficient to start modulating this GST activity. And that's exactly what became the hypothesis. Okay, we have the compound that shows already very good affinity for JAK3, but then we need to place a methyl group at the right place to then fine-tune that stability in blood. So first attempt, putting the methyl group in that particular position that is shown with compound D, it didn't work. So again, one of those things that didn't work. We did some calculations, and we showed that the energy minimized structure shown on the left of this equation is actually a structure that actually could collapse, actually have clashes with JAK3, and therefore that explains the lack of activity. Fortunately for us, putting the methyl on the other position led to this compound, 0665-1600, compound with excellent potency in the biochemical assay. It retained activity in a cell assay that is rolled in whole blood, 189 nanomolar, and beautiful stability in blood. So this is a compound that then was advanced forward. Before that, I just want to show you, we confirmed the covalent bond formation by crystallography, so we basically confirmed that. Now, coming back to the selectivity that I mentioned earlier, I show you that there were 10 other kinases that have a cysteine at the same position. So it was important for us to understand the effect of this compound in these other kinases. For this, we used first in vitro occupancy assay, and it actually confirmed that this compound was occupying some of these other kinases in that panel of 10 kinases. We later confirmed this in an in vivo occupancy study, and at the end, the better classification of this compound is as a JAK3 tech family kinase inhibitor. So this was how this compound advanced to the clinic, and there's been several reports of the studies of this compound. This is the most recent 2023, and this was done in alopecia areata. By this time, this compound had received a name, ritlecitinib, and this is a phase 2/3 study. Before I tell you about the results of this study, just a few things about alopecia areata. This is, again, autoimmune disorder that is characterized by the loss of hair. Usually the T cells recognize the follicles that, generate the hair and kill it, destroy it. And therefore, there's a loss of hair in the head, in the face, in the whole body. So different ranges of that. The problem also is that this disease impacts children, adolescents, adults. And especially for an adolescent, think about it. You're an adolescent, there's a significant psychological effect of having this issue because of your loss of hair. So I think pictures are gonna tell you everything. So treating a patient with this compound, 50 milligrams per day for 24 weeks, look at the effect, baseline to 24 weeks. Significant hair regrowth with this compound. And also important, highlighted here, had a favorable safety profile. So with this data, and along with a lot more data, so I'm just showing you just a few examples, but this data was enough to support the approval of this compound by the FDA on June 23rd of last year. So significant accomplishment for us, more importantly, because this compound was approved for both adults and adolescents, older than 12 years, for treating severe alopecia areata. Up to this point, there was not for adolescents. So this is the first drug approved for the treatment of adolescents with this disease. So as a medicinal chemist, I think I have contributed with a medicine that actually can help some patients with the disease. So I think it was very, very important to get to this point. So this brings me back to the end of this story, which is 3 stories, and just a few things to comment. I shared project 1, which again is phenotypic screening. We ended up with dropping the series and at the end dropping this approach. Some of the things that we did right and wrong, I don't know, right? It's difficult. One thing, phenotypic screening is very challenging. You don't know the target, and we're so used to working with a target that it's difficult when you don't know the target. We took certain risks, as I mentioned. We moved from to human data to mouse to go to in vivo. It didn't work. I mean, was even that biology retaining mouse? We don't know, right? So that was one of the challenges. I mentioned that also we took a hit that was transcriptionally silent, not much effect in transcriptomics. And perhaps for a disease as serious as fibrosis, you need something that has a lot more what we call horsepower or efficacy. You need something much more powerful. So difficult area. Second one, hemoglobin. For this project, we use virtual screening. And I share with Jose Luis, virtual screening is one of my favorite methodologies to finding new chemical matter. By far, it's very efficient, it's cheap, comparable to other methods, and really get you in a very good position. So I think we did the right thing. This was the right approach to find chemical matter for this project. Unfortunately for this candidate, it was terminated in phase 1 due to lack of efficacy. So it's not something that we can control, right? So we got to as a good company as we could get, but it's something beyond that. I think when you get to that point, I'm satisfied getting that point. At least we know that we got to the right compound. And finally, the third story, JAK3, we got to a lead, we were able to deliver a drug. We certainly had several risks along the way, as I show you. But I think given that we had better knowledge on the biology was useful, and we ended up with a couple, I would say, well, a couple of good luck situations that, actually we ended up with something that actually made it all the way to the end. So this is the last slide, and before I finish, just a few more things. This work would not happen without a very large team. So there's a very large team for each one of these projects, and they're here. And finally, before I finish, I want to say, gracias, Dr. Castillo. Thank you, Dr. Castillo. I'm one of those people that really had a really great opportunity working with him. I had him as my organic chemistry teacher when I was an undergraduate. Later on, I really like his, I really appreciate the opportunity to work in his lab for my undergraduate thesis work. Also, and I want to recognize La Doctora Alicia Hernandez Campos, because I always work with both of them, and I think both of them, deserve a lot of credit, at least from my part, so thank you, Alicia. But Dr. Rafael Castillo has been with me during all these years. And again, you see some very early pictures of my thesis exam. So he was there with me since the beginning. And one of the most important things about him is his support for his students. And you see that, different pictures of time when we met during ACS meetings, and especially in his favorite place, San Francisco. So without taking too long, but thank you, Dr. Castillo. He has been a great mentor, great friend, great person to have been as part of my life.
For the wonderful lecture. I think we have time for questions.
Thanks. It's a really good presentation. A question about the conformers that you showed. I think it was slide 33, if I'm not mistaken. I'm wondering, how do you determine the structure of those conformers?
It was all about, I think, QM calculations or some other calculations. Probably at this point, I don't have all the details of the calculations, but fortunately, we have within our group, we have a group of computational chemists that can, run the right calculations to give us that information. So I don't have the exact calculations or methodology that we use, but it must have been some either QM or some other type of approach to run the calculation.
So it was theoretical then? It was theoretical.
It was theoretical.
Okay.
Yeah. Oh,
okay.
yeah. Certainly. Yes.
Okay.
Yeah.
Yes.
Any other question? I think I have one. So, again, thank you so much for the very, very nice talk. In the second project, when you started doing virtual screening, at the very end, you showed that for selected compounds, you did like a complete profiling, including her. So, like, in your experience in this particular project, what about like pre-filtering the compounds from the beginning in the virtual screening? As you have seen like many posters and talks, like pre-filter the compounds based on these in silico predictions. Do you think it's a good approach?
Yes. So we sometimes use that. We certainly do, and you probably know Pfizer has done a lot in terms of structural alerts. So we certainly remove compounds that have undesirable features. So that's one set of filters that regularly is like a regular practice, right? So all the compounds that have undesirable features are removed. In some cases, we may run some either polypharmacology or specific pharmacology models to remove hits. I think in this case, we actually just use the filters for structural features. We did not apply other type of approaches to remove compounds.
Thank you. Thank you.
Thank you, Doctor, for your presentation. And I have a little question about the JAK3 molecule. How long does it take you from the point you start make the project and until you get the approval for the clinical stages?
Yeah, very good point. You probably have read some papers about the time that it takes. So this is a practical example. That's why I decided to include it, because closes this drug discovery cycle. This molecule, I think, was first registered in our collection, so right after making that compound in 2014. So a little bit less than 9 years to get to approval. So it's about probably an average number for the drug discovery. There's only a few examples like Paxlovid that probably is the record keeping for that, but typically it's in the range of 10 to 12 years or even longer than that. So this is about 9 years of advancing to all the early or late preclinical, early clinical, and then kind of late clinical, phase 2, etc. So it's a long time. And a lot of that is clinical and is actually just recruiting patients. That actually takes a long time, even though you don't think about it. It's a very time-consuming part of the life of the project, and then the discussions with the FDA that also take a long time. This compound actually got a special classification from the FDA, and it helped with kind of accelerated the process, but still, it was a long time.
Thank you so much.
Yeah.
Congratulations, doctor. The question is, a design in vitro of no necessary.
Repeat that, design. You can say it in Spanish if you want, that's fine.
¿Tiene usted in vitro o no es necesario el diseño in vitro? Porque lo tiene en vivo.
¿Para cualquier proyecto o...?
No, para el que tiene usted.
Ahorita, actualmente, prácticamente for almost every project we run right now, still we start in vitro. So we rarely go in vivo right away because of the cost. It's very expensive to go in vivo. So pretty much, I would say 99%, perhaps 100% of the projects are run in vitro. We may have more than one asset, but it's usually in vitro.
Yeah. Thank you.
Let's thank again. Fuera de contexto, para nosotros es un orgullo que Agustín nos haya apoyado durante estos 10 años, participando ya sea desde donde vive, X Simposio, o viniendo acá a la Ciudad de México. Es un egresado de la Facultad de Química, tiene una trayectoria muy larga. Se encarga del desarrollo de medidor, por decirlo así, de nuevos compuestos. Entonces, la verdad, yo me siento muy orgulloso de estar aquí y por eso es mi comentario. Muchas gracias a Agustín por presentarme.
Hola.
The last one. Okay. Rápido. Muchas gracias, Agustín, por estar aquí y por tus palabras finales. ¿Cuánto tiempo se lleva en la industria desde la concepción del problema, la resolución y de llegar a una molécula que llegue al mercado? Porque acá nos tardamos años y años y años en tener una molécula activa, pero faltan muchísimas cosas que hacer. Entonces, para que los chavos sepan, ¿cuánto se tarda eso?
Voy a hablar en español mejor. Aún así es un proceso lento, porque desgraciadamente hay muchas fallas, hay muchas cosas que uno no sabe. Depende del tipo de blanco terapéutico que uno esté tratando de utilizar. Si es un blanco terapéutico que ya se conoce, puede ser un proceso más rápido porque ya va a haber pruebas, ya va a haber otro tipo de apoyo. Cuando es lo que le llaman first in class, o sea, la primera vez que uno trabaja en esa área, puede ser mucho más tardado porque uno tiene que desarrollar todo desde cero y es mucho más tardado. Así que realmente la respuesta es similar. Digamos que puede ser que sean 10 años desde que uno empieza con un proyecto, quizás más, porque cuando registramos este compuesto ya habíamos trabajado en esto quizás 2 años, 3 años. Entonces, desde empezar, posiblemente sean 13, 14 años. Es muy largo.
Muchas gracias nuevamente y un pequeño update en el horario. Vamos a tomarnos 10 minutos más y regresamos 11:40. honor and pleasure to also introduce Professor Bill Jorgensen. I'm just going to read his very short because he has an extremely long CV. So Bill Jorgensen is a graduate of Princeton and Harvard and spent 15 years on the faculty of Purdue, and in 1990 moved to Yale, where he is a Sterling professor. Bill's research covers both computational and experiment. He has combined quantum, statistical, and molecular mechanics to model chemistry in solution. He has been a leader in computational studies of organic and enzymatic reactions in solution, molecular recognition, protein ligand binding, and molecular properties. His OPLS force fields and TIP-NP water models, as we know, are widely used in the scientific community. Bill's research group has also pursued de novo drug design, synthesis, and protein crystallography, particularly in anti-infective, anti-proliferative, and anti-inflammatory agents. He pioneered the use of free energy perturbation calculations for widespread applications, including efficient drug lead optimization. Among several honors, Bill has received the ACS Award for Computers in Chemical and Pharmaceutical Research, the ACS Hildebrand Award, the AAS and ACS Fellowships, the EQB Award in Computational Biology, the Sato International Award from the Pharmaceutical Society in Japan, the Tetrahedron Prize, and the Arthur Kopp Award. He has been elected to membership in the International Academy of Quantum Molecular Science, the American Academy of Art and Sciences, and the U.S. National Academy of Sciences. He was the editor-in-chief of the ACS Journal of Chemical Information and Modeling during 2004 through 2013, and also editor-in-chief of the Journal of Chemical Theory and Computation from his founding in 2005 through 2021. Thank you so much, Professor, for being here.
Yeah, well, thank you very much for the kind introduction. Very great pleasure to be here. I thank Dr. Lira for his hospitality and managing everything for me, and very honored to celebrate Dr. Castillo's achievements. So my first time to Mexico City, so it's also been a thrill for me to get to know the area a little bit better in your huge and wonderful university. So today I'm going to tell you a little bit about the evolution, the history of computer-aided drug discovery at first, but then I will very much focus on our recent studies, including the work we did on the COVID protease. But I think it's worthwhile to go through the history so you understand how amazing in the way the evolution has been. This is, let's see if this is, okay. So this is my research group right before COVID hit. I have two Mexican people, Patty Morales and Julian Tirado Ribs, that are in my group for many, many years. So they're always Spanish-speaking people in my group. This is our original building. It still looks the same. And we have a newer building, and my labs are on the third floor of this chemical research building. So I do have an X or Twitter account, and I'm happy to have new followers. So this is a sort of typical computer-aided drug discovery cycle, and this is my version of it, but other people have similar ones. So we start out in two phases. The first, obtaining hits for a target, and then the hard part is the lead optimization. So we begin normally with an X-ray structure for a target protein, and it often has a ligand in it from somebody else's work, And we remove that ligand and then try to discover our own new inhibitors, typically. And this is done by virtual screening. You're familiar here with that, and also de novo design. So the virtual screening is normally docking. De novo design, you use programs that are we've now called generative. So you start with a small molecule and a protein binding site, and then you grow bigger molecules. And I'll have one slide on that. And that invariably does give us micromolar hits. So these are compounds that are active in a relevant assay, like a binding assay or an inhibition assay at micromolar levels. But due to the way humans are constructed, to have a drug where you're taking a reasonable dose, you typically need to be in the low nanomolar region. So to go from the micromolar to the nanomolar is really the challenging part, and that's lead optimization. And our own work has focused heavily on that aspect, in particular on the use of free energy calculations, the FEP guided lead optimization. But we do a lot of structure building with our BOM program and energy minimization, as well as the FEP calculations. Then you do have to synthesize molecules, assay them. We try to do crystallography, and you repeat this cycle until you get to the desired potency range. There are many issues that one has to keep in mind. The issue of properties, so having reasonable solubility, permeability, avoiding reactive functional groups, everything that goes into making a drug-like molecule. And so I've developed software to help in the prediction of those properties. This was done in the early 2000s and called the QuickProp program. I'll show you one slide on that. And that's distributed by Schrodinger. It's very widely used. The last time I saw there were more than 300 users, that is pharmaceutical companies, biotech companies. The US FDA is one of the top 10 users of QuickProp, so they're interested in the predictions that we make. So that's an issue. Modeling, there's lots of issues. The choice of force fields, the convergence of calculations. Synthesis is, of course, a big bottleneck for most people. It's expensive, and the people who do it have to be very patient. You know, contrary to naive ideas that you can just design a synthesis on paper, go in the lab and execute it, that virtually always fails. And so synthesis, as you know, it requires people that are very dedicated and very perseverant. So I have great admiration for the hard work and, again, perseverance of synthetic organic chemists. Then assaying has its own issues. We can talk more about that, but I will say for anybody who is on the chemistry side and you're not maybe doing the assays yourself, it's very important that you understand the assays that your partners are doing and that the results you're getting are making sense. So let me go through the history a little bit. I'm not going to go through all of these points, but this is, of course, a bit of a personal history, but it very much follows the evolution of the field. I just threw in, as a graduate student, I worked with E.J. Corey. He's a famous synthetic chemist at Harvard, but I worked pretty much half-time with him. The other half-time I was doing chemical physics. But the half-time I worked with him, I worked on his program called LASA. It was a program, an early program for designing syntheses. And so you would enter a molecule on a graphics screen. The first graphics terminal I ever saw was at Harvard around 19, I guess it would be 72. Evans and Sutherland system attached to a PDP-1 computer. But that was the early part of organic structure graphics, was really developed by Corey. But that project, you would draw the target molecule, then it would work backwards, giving you routes. And this was called, at the time, a rule-based expert system. There was lots of activity in AI in the 1960s and 70s, a big topic in computer science, but in chemistry too. So Corey's program and related work by others is absolutely hardcore AI. So AI is not new at all in chemistry. So I started my independent career in 1975 and worked on another AI program called Cameo, which was designed to, if you knew what was in the pot, the reagents and conditions, could you predict what would come out? And this was also using rules, a rule-based system. But at the same time, I was very interested in doing computational studies on chemistry. Initially in the gas phase, I started out as an undergraduate doing quantum mechanical calculations, but then I could see the importance of solvation. So there's tremendous solvent effects in organic chemistry. Biology occurs in solution. So this was at the very beginnings of computations on fluid phase systems. So in the 1970s, we didn't have the hardware. We didn't have software, programs like Amber, Charm. They were developed in the 80s. And then, so anything you did here, you had to do, write your own code, which I did. And I wrote software to do Monte Carlo simulations of pure liquids. Then over time, that led to the development in 1983 of the tip 3P and tip 4P water models. So these are, so this is things I did myself. But by that time, we had enough computer resources that you could do iterative simulations on water, like 125 molecules in a box. And you could modify the parameters and then optimize to reproduce experimental heats of vaporization and density in particular, so key properties of liquids. Prior to that, we didn't have the computer resources, so the prior water models disappeared because it turned out when we could do this type of calculation in the NPT ensemble, the early water models gave very poor densities for water. You can't get density by chance. It's very sensitive to the Leonard-Jones sigma parameter. So that's how the tip water bottles came about. And they're still the most wide, tip 3P is still very heavily used by the Amber and Charm communities. So then FEP, when did that start? Well, there were no FEP calculations on molecular systems before 1985. The little bit of free energy calculations, I'll show you that, I was using a thing called umbrella sampling. But I have another slide on that. But there was no FEP before 1985. And it wasn't called FEP in 1985. It was called SPT, Statistical Perturbation Theory. So this goes on, and without water models and without the software, it's hard to do simulations of proteins in water. So when did that start? Well, that started around 1985, and people, myself and Julian Tirado-Rose, did early calculations on a small protein, not BPTI. This happened to be an ovomucoid, which is another protease inhibitor, but it's the same size, about 65 residues. We had maybe 1,000 water molecules for 100 picoseconds. That was done in 1988. And so that would be 100 picoseconds on a tiny little protein was all you could do in 1988. Then how about protein ligand binding? Well, that didn't start for a while. And what people were doing, though, in the 80s, or the late 80s and into the 90s, was studying organic host-guest systems and computing relative free energies of binding for those. And here are some of the ones we had worked on, so calixarenes, tweezers, and cyclophanes, etc. So that was the type of thing that was going on until the 90s. And then the force fields, the currents or versions of Amber, Charm, and OPLS are all atom. Those were developed in the 1990s. Prior to that, we were using united atom force fields, so hydrogens on carbon were implicit. Well, I will skip ahead here a little bit. So when did we really start doing protein ligand binding. Well, we did FEP calculations on that. I modified, with Julian's help, the BOSS program, or Monte Carlo program, to work on proteins. And so by 97, we were doing FEP calculations on proteins, but there was no, there was no prospective work being done at that time. We're still a long way from using FEP calculations in ligand design. So this goes on. I do mention around 2000, early 2000, there was a big uptick in interest, in part due to Lipinski, Lipinski's rules. That was in the early 2000s, where there had been a lot of failures in clinical trials due to ADME problems. So Lipinski at Pfizer stirred up the pot there, and that was the time when I wrote the QuickProp program. And I'll just show you one slide of that. And then for de novo design, I wrote this program called BOMB. So just to the students out there, I was and still am a very active programmer. Okay, so that without the programming, I would have no career. So it's really the development of the software has enabled me and my group to do things before other people have been able to do them. So I had to code up the FEP calculations in my Monte Carlo code, and I just did that before other people got to it. So when did we start the FEP-guided lead optimization? Well, that really started in 2004. At that point, I added people to my group who did experimental work. It's very hard to get people to do a lot of synthesis for you. As I mentioned, it's hard work, okay? So I recognized that, and I then I was able to hire synthetic people, and we started then doing the design and the synthesis and some assaying, but I also then had an important collaborator in our medical school, Karen Anderson, who was helping with assaying, and our initial work was on HIV reverse transcriptase. Then you might wonder about Schrodinger. So I've been affiliated with Schrodinger since their beginning. I'm on their advisory board, though we're not very active advisory board. But their force field's called OPLS because I gave them our OPLS force field, and then they worked from that to make their version, so their current version, like OPLS 4. So I call my force field, which is public domain, OPLS AA. Theirs, which you need their agreement with them. It's OPLS 3, OPLS 4. Also, I got them interested in FEP calculations, and their initial work with FEP calculations, they took my MC Pro program and put the front end on it, Maestro, and that was in 2006, and that they called MC Pro Plus. It wasn't until 2015 that they had FEP Plus. So FEP Plus is the program that is used the most in the pharmaceutical industry for doing FEP calculations. In academia, you do see other software, Gromos and NAMD, Charm, Amber being used. But FEP Plus has been revolutionary for the adoption of FEP by the pharmaceutical industry. Okay, then other things, just for those who are interested in our force fields, we have this LigParGen web server that's very heavily used by people all around the world. And then the projects that we took on with our FEP-guided work included HIV reverse transcriptase. We've worked on various kinases, MIF, etc. And then in 2020, when COVID hit, we, as I will show you, got very quickly involved in developing protease inhibitors. Okay, so that's a little background, and you can see there was nothing prior to the early 2000s. It was prospective work on using FEP calculations for lead optimization. So here's just one slide on QuickProp, since there's so much interest in AI. And I take QuickProp is quick. Okay, you can run it some microsecond because I had trained it on experimental data for different properties that I collected in the early 2000s. And so it takes an input structure So here's fentanyl, for example, and you process it. It'll take it in any form you want, a mole file, PDB file, Z matrix. And then it, first of all, it does a pattern recognition to predict metabolites. So it says there are 4 likely primary metabolic processes here. So this is handy. What we find is there are very few drugs that have reasonable bioavailability if they have more than about 6 or 7 metabolic processes that QuickProp identifies. What we can't predict here is the kinetics. So it could be that one metabolic process is enough to do you in. But empirically, we find that the more of these you have, the more likely it is you're going to have problems. But it does point out the things to look like. So parahydroxylation of an aryl. So here, you get an OH, here an OH, etc. Rings like this, they get hydroxylated and ring open. Then, using regression equations, and also some machine learning, because I keep the databases I train it on there, so I can do some, you'll see I have a log P prediction here. I have 2 of them. I have an algorithm that predicts a log P of 4.26. What's in the parentheses is the range that I observed for known oral drugs, just as a reference. And I also have a thing called log POW by 99% similarity. This, I never published any of this because I just, I thought it was too, in a way, straightforward. But the similarity thing was cute. So what I did is I keep the database, and then I compare the input molecule to molecules in the database. And so I do a Tanimoto score based on descriptors that I compute that are the same descriptors I use in the regression equations. So for this molecule, fentanyl, it tells you which are similar molecules in the database. There are about 2,000 drugs in the database. So this one, cloparastine and encanide, are claimed to be similar, and I think you can see some similarities there. So that's kind of fun. But then I can merge the data, the actual data, with my prediction from my unbiased regression equations, and the closer I get to a known compound, I weight it in. So there's a compound by 99% similarity means I must basically have the experimental data for fentanyl, and that's 4.06. All right, so solubility, same thing. I have a prediction and also one by similarity. And when you get consensus, you feel good, and I'm pretty sure that's what the answer is. We predict other things, serum protein binding, log BB for blood-brain barrier penetration, qualitative prediction of CNS activity plus, as you know, that one's plus or plus plus. hERG channel prediction says this may be a hERG channel blocker. And Keiko permeability, this compound's predicted to have high permeability, and overall percent human oral absorption says this is going to be very high. So this is all done early 2000s. I've updated it a little bit, and you can add to the databases. But this was a unique program at that time because it predicted so many things. The contemporary programs at the time was like C log P, and all C log P does is predict log P. Okay? But I did this out of necessity because in our own drug design work we were going to use it. Then this is the BOMB program. So the BOMB program is a generative program, and you can build libraries of molecules unbound to a protein, but you can also build them bound to the protein. So this is a protein called MIF. Whoops, sorry, I gotta go back. This is a protein called MIF, and just some of the residues. And the way the program works is you start with a core, just like a medicinal chemist starts with a core. The program is meant to think like a medicinal chemist. So I've taken phenol, and the user positions it in the binding site, and he's got a hydrogen bond to this asparagine. And then the program has 800 stored substituents, all common ring systems at different substitutions, small substituents. So you can build anything you want. And it also builds them in different topologies. So you can add 4 things at a time. So you could have a ring and then sprout 4 groups off it. Or you could take a ring, sprout a ring, another ring, and sprout 2 groups off that. So 4, you can make 4 additions in different topologies. Then, of course, you can take any of those and do it again if you want. And so the program does some optimization using a force field so that the ligand structures bound protein come out looking very nice. There's also a thorough conformational search that's done. And so you can ask it to build molecules. So here we've added 2 rings together. That's a 5-membered ring and then a 6-membered ring that fit the site well. But you build libraries basically with it based on the core. Okay. So that's, again, a growing program. There are earlier ones. The program LUDI is one of the earliest growing programs. This was from Hoffman-LaRoche. Okay. All right, free energy. So here's some famous old fellows. Gibbs. So Gibbs and Kirkwood here were both at Yale. So Gibbs was an early faculty member at Yale. He again died before he could be given a Nobel Prize, so 1903. And he is the father of all things free energy. Kirkwood was a professor at Yale who unfortunately died, as you can see, at a young age, just 52 years old. And but he is famous for being a stat-mech person, and Kirkwood integration is associated with him, which is that you can compute the free energy difference between a system A and a system B by integrating the derivative of the potential energy with respect to a parameter lambda that converts A to B. So this could be any type of transformation. Where we're using it with drug discovery is we're like adding a methyl group. So we go from benzene to toluene. Lambda equals 0 is benzene, lambda equals 1 is toluene. Then statistical perturbation theory goes back to the English physicist Peierls, who actually received a Nobel Prize, not for this, but for other things. And you can see this equation in Landau and Lifshitz' book in 1951. They attribute it to Peierls. And it is a perturbation where, again, you're evaluating the free energy change from A to B as a function of the difference in the potential energy and its fluctuation. Then Zwanzig was a postdoc with Kirkwood, okay, so also from Yale, and he published the Zwanzig equation, which I think was known to earlier people, but he wrote it down because the Peierls equation is just the first order expansion of the Zwanzig equation. Then next, I'd say, big event was umbrella sampling, which was 20 years later, 1975. I won't go into the details of that, but again, what it means here is you can run a simulation for A, but then pretend that you're B, and you can have a biasing function that lets you sample A and B simultaneously, and you can get the free energy change from that. That was used in the early free energy calculations that were done in the late 1970s by the group of Bruce Byrne and also by my group. And so those calculations were simple things like butane in a Leonard-Jones liquid, okay? So what you're doing is calculating the change in the gauche anti-population for butane in the gas phase versus in a Leonard-Jones liquid. So it wasn't until 1982 that Byrne and I got around to butane in water. So that's the status of things in 1980. This is state-of-the-art calculation. 1982 would be an umbrella sampling calculation to study the conformational shift for butane in water. And we both agreed that there was a shift where you get a little more gauche in water than you get in the gas phase. So that initial feeling of a hydrophobic effect, making butane fold up a little bit. But that was where we were for free energy in 1982. So 85, so I decided to do the FEP calculations because I had looked in the Macquarie book, and there was also a little discussion of Zwanzig's paper. In 1984, Zwanzig's paper has 8 citations. In 1985, it has 9 citations. So this was just obscure stuff, right, at that time. But I decided to give it a try for a molecular mutation, methanol to ethane in water, and it worked out incredibly well in that we could compute the free energy change very precisely. The uncertainty was on the order of 1/10 of a kcal per mole. That was what was really revolutionary. It also had good accuracy because the force field, you got the right result, but the really important thing was the precision. So now we could compute free energy change very precisely. Well, so you can imagine that the excitement that nobody experienced with this, because they found out that it took me 6 weeks on our only computer to generate this, okay? So the calculations require you to do a series of simulations, so it was very time consuming. And back then, in my lab, we were really, really unfortunate. We had a computer. We had a Harris 80 computer. So we had one processor. The Purdue Computer Center at that time, I think, had 3 processors for a university with 40,000 students. Literally 3 processors. This laptop probably has 8 processors, right? And they're way, way faster. So you just didn't have the computer resources, and that, held things back. But there were certain people, Peter Coleman in particular, he's a very old friend of mine. He was an undergraduate in the same lab I was an undergraduate in the lab at Princeton where he was a graduate student. So we were always very close. And Peter got the significance and off to the races on that. So for now to lead optimization, so it was recognized at that time, the mid-'80s, that you could do calculations for ligand design with FEP. You can also do it with thermodynamic integration, but FEP is a very convenient way of doing things. And here is the typical cycle. You have red protein, green ligand to give complex, red protein, blue ligand to give complex. So in the computer, you evaluate green to blue. That's just the ethane to methanol calculation. But then you'd also do it in water, but you do it for the complex. So this lets you then, by difference, compute the change in free energy of binding between A and B. And that's just what you need for lead optimization. So we just try different things and see what gives you improved binding. And so we run the calculations, many others. You have 1 protein, 1 ligand, and a couple thousand water molecules in a ball. And we're doing Monte Carlo. We also do MD sometimes. So the type of question you can ask is this. You can optimize substituents on a ring. This is a schematic of Celebrex bound to the COX-2, a binding site. And we can ask, what happens if we change the methyl group to other substituents? And this was known experimentally, and we can compare the calculated with the experimental. So this is the type of thing we were doing in 2000. This paper was published in JACS. And what, of course, we've got here, we had very good agreement between computation and experiment. That's a hydrophobic pocket. We're just making little changes, and all is good. But there's nothing predicted here. This is retrospective, and it's always worth having a little grain of skepticism when theoreticians are doing things that are purely retrospective. Okay, so the question is, can you actually predict something? So that's what we dedicated the next 25 years to. So what happened was, in the 80s, we all knew, the community, that you could do this in principle, but then when people tried it, it didn't go very well because of the computer resources. We also, the force fields weren't as good as they are today. And I'd say the 90s were a lot of duds. Some people thought that FEP had been overhyped. So where we are today with AI, I guess. And then the 2000s were pretty lonely, but we hung in there and we were working on HIV reverse transcriptase. And fortunately, by the end of that decade, we had enough very favorable results that we had convinced Schrodinger to pursue this, and we could publish this accounts article. So the way it works is simply like this. So the key to success is making small modifications to substituents, linkers, and rings. So this is HIV reverse transcriptase. And so some of you are familiar with this. There are two types of inhibitors. Like most HIV drugs have targeted HIV reverse transcriptase. The nucleosides like AZT, DDI, DDC, and then the non-nucleosides like nevirapine, efavirenz. So the original one-a-day pill was a combination of two nucleosides and one non-nucleoside. We've tried to make better non-nucleosides. So here's the non-nucleoside binding pocket. It's an allosteric site. It's a very rare case. There are very, very few drugs that actually bind to allosteric sites. This is a key exception. And this is a compound, the EC50s here in a standard cell assay where you have T cells, human T cells, infected with live HIV virus. So this is done in a BL3 facility. My colleague Karen Anderson's lab in our med school does that for the compounds we design and synthesize. So this is a de novo compound that we had cooked up with the BOMB program. And when Karen assayed it, it was 10 micromolar, very small compound. Then the point is, if you happen to know to put a cyano group here, you could get a 50-fold boost. That little addition gets you to 200 nanomolar. Then if you happen to know to change the thiazole to a pyrimidine, that gets you to 17 nanomolar. Then if you happen to know to put a methoxy group there, you're at 2 nanomolar. And this is the crystal structure of that 2 nanomolar compound. We got much later than when this work was done in 2006. The point is you need to form systematic small group linker and heterocycle scans. So what you do here is you see open sites like this and we scan in a chlorine and a methyl. If chlorine works out well, then we'll try something that's a little more withdrawing like cyano. We certainly go through standard series of monocycles containing heteroatoms, and that would bring you to the pyrimidine. You rescan, you'd see a methyl group is good here, so then that, to do a little better, a little more donating, you put in a methoxy group. So by a series of FEP scans, you can get to this 2 nanomolar compound, and that's exactly what we did. Okay, so here are two of our earliest papers, 2006. The first one showing that series of compounds where we're scanning for different R groups. But I'll just highlight the second paper. This is a JAX communication from 2006. And there are 3 compounds. So here we were trying to change the heterocycle, and we went through monocycles, but we were then interested in bicycles. And you can cook up all sorts of bicycles. Hopefully you can synthesize them. But I'm going to show you these 3. These are isomers. So this is a, you can just see the NH is there, but I can move it 1 click over to there, or I can move it another click over to there. So they're isomers. And it's pretty tough if you even have built the structures to try to predict which one of these is better than the other. But there's the results. So by FEP, we had predicted that 9 was the worst, and then 10, and then 11 was the best. And then when they're assayed, again in the cell assay, you can see you go from 900 nanomolar down to, I think it's 19 nanomolar. So this is the power of FEP. It lets you explore things that are definitely not obvious just from building structures. So for HIV, we published many papers using the FEP design. Here's that early 2 nanomolar compound, 2006. Here's the one I just showed you, another 5 nanomolar compound. And here, now 5 years later, we have this more complicated compound, this 2.5 nanomolar. Now, why would we be doing that? Okay, why didn't we stop in 2006? Well, as you all know, viruses mutate. So what I'm showing you here is the potency against wild-type HIV-1. But HIV mutates rapidly, particularly when treated with these drug classes, and this compound is not active against very important mutants. Y181C arises quickly in patients who are given drugs of this type. This compound is 2.5 nanomolar against wild type, but it's 5 nanomolar against Y181C. So we're constantly here, we're trying to make a broader mutant spectrum. So you want compounds that will work against all clinically observed mutants, and that's been the challenge with HIV. But we've had other, I'll highlight that compound. So this is now a compound that came from docking. And so it's a 5 micromolar docking hit. So that we evolved. Initially, a key discovery here was to put an oxygen in. That changes the orientation of the 2 benzene rings. And that, with a little additional modification, brought us to sub-nanomolar compounds. We had a compound, our most potent compound in this series is 55 picomolar. This is a 320 picomolar compound that's 85 nanomolar against a very difficult double mutant, K103NY181C. And this compound, now we have cyclized the cyanovinyl group into worrying about its microreactivity, though there's an important drug in this class, approved drug that has a cyanovinyl group, rilpivirine and etravirine. But we cyclized that and picked up this compound, an indolazine, not the most obvious heterocycle, but we did, as I'll show you, a scan of heterocycles, and that compound is, I can't quite read it, but it's sub-nanomolar on wild type, and it's also about 10 nanomolar on the double mutant. So that's a great compound. And we've done other things. You can see we even have covalent inhibitors. But let me just show you a little more in that cyanovinyl group. So what you worry about with cyanovinyl groups is that you could have covalent modification and off target, including glutathione. I think Dr. Garcia mentioned glutathione. If you diminish your glutathione, you end up with liver toxicity. So what are their drug precedents with the cyanovinyl? And the answer is very few. And some of them I'd say are dubious. They're really tautomers. So this and this. And the one additional one is, I don't show it here, but rilpivirine. So cyanovinyl is very rare in drugs. So we wanted to get rid of it, and we got rid of it by incorporating it into a heterocycle. So here are then, the problem is you have many possible heterocycles where we can have a carbon here or a nitrogen here. And if you build them in the binding site, you can see that this edge is in a aryl-aryl interaction with TRP-229. But it's very difficult to eyeball what would be the best bicyclic heterocycle. So this is where, again, FEP to the rescue. So many options with unclear interactions. So we looked at all of these 18 alternatives, just the core structures there, and the numbers in parentheses are the relative free energies of binding from the FEP. And that highlighted having this indole, the indolazine, and that benzofuran, but not the isomeric indolazine or benzofuran. So off we go to synthesis, and we synthesize compounds in these series. We also can synthesize a couple reference compounds in the isomeric series, and the answer is exactly correct. In each case, these were more potent, and we got some pretty good compounds out of that. So this is a 10 nanomolar, the indole, 10 nanomolar, but it's 800 nanomolar on the double mutant. The benzofuran was 7 nanomolar, but non-active in the double mutant. But this one, the indolazine, is 380 picomolar and 11 nanomolar on the double mutant. And we never would have gotten to the indolazine without, I didn't know anything about indolazines before we started doing this type of perturbation. We wouldn't have gotten to that compound without the FEP calculations. We have many crystal structures of these compounds. This is that specific compound bound to HAVRT, and there's that edge-to-face interaction with TRP229. We know some reasons why it's better in the double mutant. It's pushed farther back into the pocket, and that helps it get away from K103 over here. So we have published on that if you're interested. And then this led to our lead compound, which is a very similar compound, but now with a naphthol, and we call JLJ636. We have crystal structures for that. We have PK data, and here it is in comparison to efavirenz, which was in the first one-a-day pill, a tripla. So this is a DuPont-Merck compound. Dose 20 mg per kg. This is Cmax, is much higher than the efavirenz. They both have the same Tmax. We have much higher area under the curve, 10 times higher, and clearance, very, very favorable numbers there, slow, and we have no cytotoxicity in the T cells, whereas this is 15 micromolar. This compound has also been in different formulations, so in nanoparticles and implants. You can give a mouse one dose of this, and it will, in an implant, and it will provide protection against HIV infection for a month. So an extended release idea. So that brings me to COVID. So we had done a lot of work on viruses, and my colleague Karen, has the BL3 facility. And in January 2020, there's a lot more interest in COVID. Then by March of 2020, we were shut out of the lab. Okay? And we were out of the lab until June 15th. But of course we can compute. And so I said to my people in my lab and to Karen, I said, I think we should give a shot on COVID, so let's learn a little bit more about it. And COVID and HIV are both RNA viruses, but their insides are quite different. There is no reverse transcriptase with COVID, but there is a protease. There are two proteases, but the one we've worked on is the main protease. So this is the life cycle. I won't dwell on that. But this is the protease, and if you can shut down the protease, you shut down the reproduction of the virus. So simple as that. And there was a crystal structure available. It's a dimer. So it's called MPRO, the main protease. So we decided we would go after it. So what does that mean? Well, we have to get some initial hits, and we decided to do docking. And not only did we do docking, I decided that we should dock known drugs, with the hope that you might get a hit that would show some activity against COVID. But I was really looking for an initial starting point for then doing further lead optimization. So Karen got the plasmid to express the protease. So there's nothing infectious at this point. We just have a protein. There's no cells. We just have the isolated protein. We don't have the virus. And there was in the literature, there's a standard FRET assay available so that you can detect the cleavage of a peptide substrate. So Karen's lab set that up. And then I asked Julian and Mohammed and others to do docking. And I keep a database of 2,000 known oral drugs. I've mentioned it already, and I update it. And I said, dock the drug database and do consensus docking. So we do consensus docking. We use Glide, Autodoc, and Autodoc Vena. But we have 4 protocols. So we dock the 2,000 compounds, and we get the scores against the MPRO. And then we look for the ones where you had 3 of the 4 scores were very favorable, below a cutoff. And there were about 100 of those. And so I then looked at them individually. This is called Bill's score. So I'm going to score them myself because there's no scoring function that's all that great. That's why we do consensus scoring. And I always picking up little things I don't like in the poses. So the pose is what you call the picture. This is a pose here. The picture of the protein ligand complex. So I spend a day or two just popping up 100 structures on the computer screen and rotating them, looking at them, and deciding which ones I like and which ones I think have been misscored. And that led me to tell the folks to purchase 17 compounds. And remarkably, when Karen ran the assay, this is now in June, we had done the docking, but then the experimental work really started in June of 2020. We found that 14 of the compounds showed some activity. So this was a miracle. Usually you don't have that kind of success. And the most potent compounds were around 4 or 5 micromolar. So we then did an IC50 rather than just percent inhibition. So that was interesting. We published this paper. And this is a docking pose of a compound of azolastine. So now what I didn't say is the compounds we're most interested in. So looking at all these, one most interested also involves synthesis. So which compound can I take that I think I can make analogs without much trouble? So azolastine here has a stereocenter or 2 and I think a 7-membered ring. And so that wasn't my favorite compound. My favorite compound was a relatively weak compound called parampentyl. So it is in our assay, it's in around 150 micromolar. It was kind of amusing when I sent in this BMCL, I think. Well, no, it was ACS MedChem letters. So we sent in the ACS MedChem letters paper with the docking results. And I got back some sort of snarky review from one person who said, well, most of these are just useless. I mean, they're just too weak to ever be useful at all. Maybe there's maybe 1 or 2 of these that is worth reporting. I'm sitting there chuckling because we then took the azolastine, which was one of these useless compounds, and evolved it into what you'll see is low nanomolar protease inhibitors. So I liked this because I could see how to synthesize it. So, we use these aryl systems you make by Suzuki Chemistry or Chan-Lam couplings. And so, yeah, I can make that. No stereocenters, check, that's good. It's a drug, so that's good. It's a good starting point. And I could see this is the Glide pose. Glide generally gives good poses. And I could see features that weren't optimal. So this nitrogen in the pyridine ring is just pointing into solvent. That's not doing any good. There's a carbonyl here, but it wasn't hydrogen bonded to anything. But over here, there's an NH that would like to have a hydrogen bond. So maybe I could flip that over there. There's a histidine here that would like to be in a hydrogen bond, but that benzene ring doesn't do it. So maybe we could change that to a pyridine. There's also a little space, so maybe I can grow something there. And I could also see this edge might, if I modified it into a heterocycle, maybe I could pick up some hydrogen bonding over here. So with those ideas, then we do FEP. And these I ran myself. Like I told the students back there, I'm not completely useless at this point. So I ran these myself because other people had other things to do. And this is my high-tech data storage system called Excel. And I'm showing here, these are 2-pyridyl, 3-pyridyl. So I'm changing that ring on the left from a benzene ring to these different heterocycles. And I have the bound and the unbound. The difference is the delta-delta G. And the ones I'm really interested in is where I have a nice negative number. And that was for 3-pyridyl, which I'll show in a second. This is the CO flip, where I was flipping it to pick up that hydrogen bond. That was predicted to be dynamite, -4. And then I did a chlorine scan on this benzene ring, which said, yeah, there's room, and try putting in a meta-chlorine. So that led to then the first compounds that we synthesized. So there are only 2 synthetic chemists here, a postdoc, Chun Wei, and a graduate student, Lizzie. And so I asked them to make these 3 compounds. And then this was the result. So our 150 micromolar compound very quickly became a 10, a 6, and a 4 micromolar compound. So don't ever tell somebody that a 150 micromolar compound is useless. Okay. So in boom, and this our first 3 compounds were down at 4 or 5 micromolar. Plus we had 2 series because we had worked a lot with uracils, and I built in a uracil, and it seemed to pick up hydrogen bonds on that edge I was saying. And so Chun Wei made the uracils, and Lizzie was making the cyanophenyl compounds. And we had now 2 series, so then they kept making compounds. Then the next step, we didn't get a crystal structure for 2 months. So this is just all predicted by the computer. But 2 months later, we did get a crystal structure for this one compound here with the 2 chlorines, and it was exactly as we had modeled. Okay, this absolutely dead on. There's a hydrogen bond there, the one there, the cyano makes a hydrogen bond here. This is the cysteine. It's a cysteine protease. That's the cysteine, and this histidine is part of the catalytic process. So here's our crystal structure. But we had already started doing FEP because there's a channel running up here we call the S3-S4 channel. And so it was very clear that we would build the molecule this way. Here's the crystal structure, again, reminding you it's a dimer. There's our little molecule. And these are all deposited, of course. So here's some of the FEP results for growing in that channel. Just a typical medchem scan, methylethylpropyl, methyloethylpropyl, butyl, mom. And then for grins, I just added these to fill out the table. But I knew they probably weren't going to be very good because these hydroxyl ones, they're very happy unbrowned. It's hard to introduce a hydroxyl group and pick up binding. So I ran the FEP calculations, and that, as I expected, said, oh, propyl looked really good. And so we made then next, in both series, the O-propyl compounds, and boom, we're at 140 nanomolar. And then later, we made the CF3 derivatives. Okay, I don't like CF3 groups. They generally make compounds poorly soluble. So we try to avoid them. There are drugs with CF3 groups, but very often you're struggling with solubility, and CF3 never helps solubility. But that gets you to 25 nanomolar. That was, again, done later. So I did more modeling of more crystal structures. There's the propyl compound, and I knew that the propyl would have a gauche conformer at the terminus because that would put the methyl group right into this hydrophobic pocket, the S4 pocket. And so that was confirmed again 2 months later with the crystal structure. But we had done additional modeling. By looking at this, I could see I could probably put a phenyl here. So I built molecules like that. And then as soon as I have a phenyl, I'm going to do chlorine and methyl scan. That said, orthomethyl and orthochlorine is the way to go. So we started making compounds like that. And there we have the unsubstituted is 128 nanomolar, and the orthochloro compound is 18 nanomolar. Okay, so at this point we'd probably made 25 compounds. Okay, and there's another crystal structure, and we published this then. We finished this work in the fall of 2020, so we didn't start till June 15th, right? And not only this, what I'm going to show you, but we also did the antiviral assays that are in this paper. So here's the team that involved, the two synthetic people first, Karen and myself at the end. And this was published in ACS Central Science in February of 21. And we published maybe 30 or more compounds, and many of them are under 50 nanomolar. So the next issue, though, is this is just in the protease assay. So you got to show that this is going to work in infected cells. So off we go to the BL3 facility. And here is our technician, Casimiro, who's the same technician we've used with HIV, in full hazmat gear because, as you all sadly know, COVID is airborne. Well, there he is. And then we get the results for some of our compounds. And long story short, so we have assays that are done here with the infectious cells. We also have so-called replicon assay. But these are the infectious cells. And our reference compound, remdesivir, it's not a protease inhibitor, but it is an anti-COVID compound. It's basically 1 micromolar. Our best compound is number 19. It's an 80 nanomolar inhibitor. Here it is. We call it MPRO61. And we have crystal structures for that. And this is 80 nanomolar. Johnson & Johnson was very interested in these compounds, and we gave some of these compounds to them, and they ran it in their assays, and in their cell line it was 50 nanomolar. The Pfizer compound that is in Paxlovid is nirmatrelvir, and it has an IC50 of 19 nanomolar. There's the compound. The EC50, if you include an efflux inhibitor, is 740 nanomolar, so 10 times less potent than our compound, and 4 micromolar if you don't have the efflux inhibitor. The other problem with Paxlovid, I think as many of you know, is it has CYP3A4 inhibition. So to overcome that, it's co-administered with ritonavir, which is an old HIV protease inhibitor. So, Pfizer really jumped on this and got the compound approved, but it is not, I would say, you can do better. Our compound's also non-covalent. This is reversible covalent. It has a whole bunch of stereo centers. It's peptide-like. Ours is non-peptidic. You can imagine the cost of goods difference here. And then our compound has no CYP inhibition. So we've done some preclinical work with it. Some of this is from a recent PNAS article. This is a PanLabs panel for off-target activity. There's basically nothing, unbelievably clean, a little bit of hERG. And then we have the bioavailability in the rats, excellent. We also decided to look at synergy with the Merck inhibitor molnupiravir, which is not a protease inhibitor. But this is mice, and where the virus goes, after 6 days, it goes into the lungs and into the brain. They're dead. Or with our compound, it's clean for 6 days. At day 8, you see some in the brain. With molnupiravir, similar to our compound, if you take the two compounds together, they're very effective. And this is body weight for the different combinations. And the ones that are taking the combination therapy are actually gaining weight. Okay, so that's what I'm going to tell you. And I hope I've shown you the power of FEP and also where this came from. It's really unbelievable. So in 1983, we have tip 3P water. And here we are designing, COVID protease inhibitors in a very accelerated manner due to the crisis that presented to us. So on June 15th, we had perimpenyl. On August 3rd, we had the 6 micromolar compound, and we didn't get the X-ray till October. September 2nd, we're at 140 nanomolar, and then by October 22nd, we're at 18 nanomolar. Total compound synthesized up to this point is about 30. We did have a couple of, sort of exploratory things that I didn't show in addition. So I think I'm personally very proud of this work and proud of it for the sake of the community because it really shows the power of computer-aided drug discovery. The people that were working on this recent group, I mentioned Chunwei Zhang and Lizzie Stone did the synthesis here. Karen Anderson has been my longtime assay associate for cell assays. And then Julian Trotta-Rose has been with me since 1985. So he's a senior research associate and it's been essential to all of our developments in modeling proteins. So I thank you again very much, and I'm very pleased to be here in the symposium honoring Dr. Castillo. Thank you.
Thank you so very much for the very nice talk and wonderful lecture. I think we have time for questions.
Hi. Hi, Dr. William. This question is on behalf of the chemists in the room that have interest in software development. And my question is, there are two questions, like what trends are rising in drug development and software development? And what are some areas of opportunity for chemists in these regards?
So the question is, where are things going?
Right.
Well, I think they're getting better gradually, but science, as you can see, this is giving you a 40-year glimpse. It's taken 40 years to build up from tip 3P water to this rapid discovery of the protease inhibitor. So it's a brick by brick. I got to see your wonderful pyramids, right? And that reminds me of just the image of science, brick by brick. So there's not going to be some AI magic breakthrough here. It requires continual hard work on the weak spots. Okay, so a key weak spot is still the docking. The scoring of the docking is poor. The poses still are, if you look at them hard, often they're not so good. And you can even have reasonable correlations of activity, score versus experiment, but then you realize the pose is wrong. So it's actually random. There's a lot to be improved with the document scoring. There's more to be improved with properties prediction that would help. All of these things require better data. The key problem with the scoring functions is they've been trained on a mishmash of data. If you look at where they've been trained on a mix of IC50s, KIs, KBs, EC50s, all lumped together. That's not good. IC50, by the way, is a semi-random number. It depends on the substrate concentration. Ki and Kd is directly related to free energy. So what you'd like is a lot of high-quality Ki and Kd data on multiple systems, obtained in a consistent manner. Then you can train on that. That doesn't exist. Even something like solubility, which is in better shape than most, if you actually look at the data, nobody measures the solubility in exactly the same way. You think, oh, we just stir a compound up in distilled water. Nobody actually does it like that. Okay, so they're always using buffers of some sort. So there's just a lot of room for improvement on the hard part, which is collecting data that can be used to train the functions you need for neural nets or AI, et cetera. And so it's going to be slow progress. The molecular dynamics, you need to be able to run longer. So there's still run, I think the convergence now, you want to go run longer than that. You're also going to see work on bigger and bigger systems. If people want to model an entire cell, so you're going to have to go to coarse graining. There, I warn people, be very careful. Make sure you're predicting something. Not just, oh, I've run the biggest simulation that's ever been run. But I didn't learn anything from other than to claim I've run the biggest simulation. So that, there's a lot of room for protein-protein interactions, protein-protein docking. That's increasing in importance.
Thank you very much for wonderful talk. There's most of this work's been done with classic force fields, but currently there are proposals for polarized force fields, mixed quantum mechanical, molecular mechanics force fields, etc. Why these advances have been incorporated in drug development so extensively?
So the questions are, you could have better force fields. So this is the amateur molecular type force field. You could have a polarizable force field. The problem is there isn't any accepted polarizable force field. There's no polarizable force fields to demonstrate to be better than what we do with these force fields. And these force fields have been updated. So we include things like halogen bonds. We include cation pi interactions. So these were things that were missing in the earlier versions. And those are bringing in polarization and electron structure. QMMM, I've done a lot of the whole lecture on what we did there on studying organic reactions with solutions. There, the QMMM, the QM part is just the reacting system. We don't have the computer resources to do the whole system or any length of simulation where it's full of QM. And if you know QM, you really have to get a pretty high level for quality levels to get the model result. So for the time being, these current additive force fields, the current version of OPS animatronic, and then the Schrodinger OPS 3.4 are the best you can do on these large systems. Where read optimization, you need reliable results in a fairly rapid manner. So I don't have an alternative yet, but over time, maybe machine learning force fields will come on. But you have to remember, these things do pretty well. If we get organic liquids, the problem with organic liquids is dead on. We now fix things so that 3 energy vibration dead on. So a polarizable model can't do better than that. It can maybe get to it. But it just hasn't happened yet. One more.
Thank you, for Professor Jorgensen, for a really, really great talk. And really, it was fascinating, the last part of your presentation for the, starting from the lead compound, getting to your compound, I think 18 was really, really nice story. For me, the question for me is that I think now we have the capability of enumerating or creating very large number of compound modifications virtually, with thousands, perhaps millions of compounds. But at the time of going from that large amount of compounds to say which ones we want to synthesize is difficult. One of the main problems is actually potency prediction. I think it still is very challenging. I have seen FEP in applications of FEP, but it still is very time consuming to go through, those calculations. For me, the example you presented was very interesting because it seems that you were able to run a number of calculations and still being able to make slow progression in terms of optimizing for potency. What is your recommendation for doing something like that when you can really enumerate large number of compounds? But then how would you do that, right? So do you focus on small transformations at a time so that you can end up with a more manageable number of calculations?
Yeah, I think as I demonstrated, at some point you follow traditional medicinal chemistry where you have a core and you see groups off that core and you can try to do optimization on each of those groups. And so scanning heterocycles, scanning substituents, and then changing the core and getting it into a different series. So there's nothing new there, but the user needs to be careful with picking the core. That's critical. And that also is where knowledge of synthesis comes in, where you want to pick cores that you're comfortable with synthetically. But it's just as I indicated, you want to think about all the time optimizing substituents, linkers, cores, and heterocycles, and doing these systematic scans. I do scans in other things. Very often with a benzene ring, you'll see it substituted as two substituents, but not with more off the core. So we'll do a double scan. So like for benzene with 5 potent sites, if you put in 2 chlorines, I think there are 11 ways of doing that. So we go into all 11 in parallel. And then that will tell you the best pattern to substitute is like 2,6 or 3,5, etc. So you can do that too. It's just being systematic. Substituents, linkers, heterocycles, scans.
Thank you.
Let's thank again Professor Jorgensen for the very nice talk. Thank you. Now I'm going to switch to Spanish. Vamos a continuar con el programa, vamos a hacerlo ahora en español. Y nuestra siguiente ponente es la doctora Alicia Hernández Campos.
Vamos.
Okay.
Hola, buenas tardes. Bueno, pues ahora toca la parte emocionante de este simposio. Doctor Castillo, bienvenido al simposio que lleva su nombre, Rafael Castillo. Muchos de ustedes ya lo conocen y lo conocen en su mejor versión. Muchos no, pero el doctor Agustín nos va a hacer un breve resumen en atención al doctor Jorgensen para dar un pequeño resumen o una semblanza sobre su trayectoria. Adelante, doctor Agustín, por favor.
Bueno, va a ser la semblanza en inglés una vez para facilitar que el doctor Jorgensen pueda entenderla. Así que voy a empezar con el doctor Rafael Castillo Bocanegra was born in Villahermosa, Tabasco. He completed his studies in chemistry, pharmacy, biology, or QFB, at the Facultad de Química, UNAM. He then pursued graduate studies at the University of California, San Francisco, where he obtained his PhD degree in pharmaceutical chemistry in 1977. His PhD thesis title was Synthesis and Biological Activity of Farnesyl Pyrophosphate Analogs, under the guidance of Professor Paul Ortiz de Montellano. His academic activities started at Facultad de Química UNAM in 1970 as an assistant professor. After completing his graduate studies, he returned to Facultad de Química in 1976 as a full-time professor in pharmaceutical chemistry. Since then, and for over 45 years, he has taught without interruption, except in 3 periods, while being in sabbatical leave, the semester-long organic chemistry course level 1, 2, 3, or 4 for undergraduate students, the semester-long pharmaceutical chemistry course for the master's in pharmacy, and later, which was later the master's degree in chemical sciences. Throughout his professional career, he has demonstrated a strong academic training and a noticeable capacity to transfer knowledge in his area of specialization, organic chemistry and pharmaceutical chemistry. Since he joined the Facultad de Química, he has worked to promote graduate studies in pharmaceutical sciences. He was coordinator of the Master in Pharmacy program, where he taught the pharmaceutical chemistry course as the basis for the design of new drugs. His graduate studies were focused on the philosophy of the rational design and synthesis of new drugs, more specifically in finding new inhibitors for an enzyme called esqualane synthase that he would be used to better understand the function of this enzyme, and with the long-term goal of finding new agents for the treatment of cardiovascular diseases, one of the main causes of death in the United States. With the same research philosophy, and as part of the pharmacy department at the Facultad de Química, his research line has been focused on the design and synthesis of new antiparasitic drugs, anthelmintics, and antiprotozoal drugs. More specifically, in benzimidazole analogs, a research line that he has pursued for over 35 years. More recently, he has explored the identification of inhibitors of an enzyme called AKT as a potential anti-cancer agents. Parasitic diseases and cancer are among the main public health problems in Mexico and Latin America. Always keeping at the front of research in his area, he has also pursued computer-guided drug design. He's a pioneer in the application of these methods for the design of new antiparasitic drugs. Dr. Castillo's research has established the relevance of different substitutions patterns in the benzimidazole ring for antiprotozoan activity. The impact of these contributions in the area are reflected in the high number of citations to his first manuscripts in this area, published in Bioorganic and Medicinal Chemistry Letters in 2001 and 2002. Given the multidisciplinary nature of his research, he has established several collaborations with both national and international institutions throughout the years, leading to the formation of research groups that include well-known researchers in the area. As a result of these collaborations, important structural requirements for the antiprotozoal activity of benzimidazole analogs has been established, including for Giardia intestinalis, Trichomonas vaginalis, and Entomoeba histolytica, protozoans of high incidence in Mexico, for Trypanosoma cruzi and Leishmania mexicana, protozoans that cause diseases that have received little attention in the world, and for anthelminths for Fasciola hepatica, Taenia crassiceps, and Trichinella spiralis, where his research has also been focused in establishing potential mechanisms of action for the activity of these compounds. Another important contribution from Dr. Castillo's research is the discovery of triclabendazole biosaustery. Triclabendazole is the drug of choice for the treatment of the early and adult phases of the parasite Fasciola hepatica. Resistance to this drug has been observed in some parasite strains. This biosaustery, called compuesto alpha, show activity comparable to triclabendazole, but also show low aqueous solubility. It was actually dosed as a suspension when this compound was tested in cattle, where it was shown to be safe, non-toxic, and non-teratogenic, but also with limited residence times in the animal. Compuesto alpha was shown to eliminate the parasite spikes that allow it to attach to the host. Additional studies using triclabendazole-resistant Fasciola hepatica strains confirm the same effect of eliminating the parasite spikes, but additionally, it eliminated spermatogenesis and destroyed the parasite's reproductive organs. Research focused on the identification of water-soluble analogs led to the disodium salt of a phospho-oxymethyl benzimidazole derivative as products for compuesto alpha and triclabendazole. Both agents were formulated as injectable solutions and demonstrated efficacy against Fasciola in sheep. They show over 88,000-fold better solubility than the original compounds. They are hydrolyzed by the enzyme alkaline phosphatase in seconds. This work led to applying for a patent in 2014, and for this accomplishment, Dr. Castillo received a first-place award for the program Fomento al Patentamiento y la Innovación, or Program for Promote Patents and Innovation, from the Coordinación de Innovación y Desarrollo de la UNAM. This patent has been pursued internationally, and it has been granted in Mexico, United States, Colombia, and Brazil. Throughout time, and with the contribution from his undergraduate and graduate students and collaborators, Dr. Castillo has accumulated a compound collection of over 450 compounds, many of them with nanomolar activity. These compounds are the result of applying the concepts of pharmaceutical chemistry. His research has led to over 135 publications in indexed national and international journals and 4 granted patents. His research has received over 1,600 citations that demonstrate the impact of his research. Moreover, he has contributed to the training of many students, including 29 undergraduate students, 60 master's degrees, and 21 doctoral degree students. Many of them are now researchers within UNAM and other national and international institutions. He has also trained postdoctoral fellows and has received students from international exchange programs. He has always put the same effort on his research and his teaching duties. In fact, one of the commitments when Dr. Castillo joined the Facultad de Química was to establish a pharmaceutical chemistry laboratory. This made him a pioneer in the area of pharmaceutical chemistry in Mexico. As mentioned earlier, he has taught the organic chemistry course to undergraduate students without interruption since joining the Facultad de Química. In addition, he has also taught courses on the synthesis of pharmaceutical agents, number 2, pharmacology number 2, selected topics in pharmacy 1 and 2, heterocyclic chemistry, and pharmaceutical chemistry 1 and 2 for graduate students. He has always encouraged his students to present the research in national and international scientific meetings, and to pursue research visits at international institutions so that they can acquire a broader experience in the area. All this has made Dr. Castillo one of the most loved and admired professor researchers in the pharmacy department. Dr. Castillo has also contributed to the institutional and administrative activities within the school. He has been part of many committees and has played important academic and administrative roles. He has been head of the organic chemistry and pharmacy departments, a member of regulatory committees in FE Zaragoza, Facultad de Química, and Instituto de Química, UNAM, a member of advisory academic committees like DIGAPA, PAPIT, for technological innovation, and in the Committee for Nomenclature for the Mexican National Pharmacopoeia. His contributions to the teaching and research have been recognized with several awards, among them Leopoldo Río de la Loza in 1986, Etelvina Medrano in 2004, Martín de la Cruz 2005, and first place for the patents and innovations in 2014. Huchimán de Plata in 2019, Academic Excellence in 2016 from the UNAM, and the Premio Universidad Nacional de Docencia en Ciencias Naturales in 2018, and Level D in the program from Primas al Desempeño del Personal Académico del Tiempo Completo de la UNAM, and from the Sistema Nacional de Investigadores, Research Level 3 for several years. With great charisma, humility, kind and respectful, passionate for his work, creative and innovator, with a great sense of humor and with a great patience for his students. Without a doubt, Dr. Rafael Castillo represents the indisputable figure of an academic researcher from the Facultad de Química and from UNAM.
Muchas gracias, doctor Agustín. No lo presenté, perdón, pero ya lo habían presentado esta mañana. Es hijo académico, como su servidora, del doctor Rafael Castillo. Ok, vamos a continuar con el programa y le pido por favor a la doctora Isabel Aguilar, que ella hasta el año pasado fue miembro de nuestro departamento de farmacia, y que ahora también goza de las mieles del descanso, de la jubilación, viene a platicarnos un poquito acerca de su acercamiento o del conocimiento como persona y académico que tuvo con el doctor Castillo durante muchísimos años. Isabel, adelante.
Buenas tardes a todos. Buenas tardes, doctor. Doctor Castillo, buenas tardes. Es para mí un gusto estar aquí en mi querida UNAM. Escribí esto para que no se me olvide ningún detalle, pero me lo sé de memoria. No es cierto. Es para mí un gusto estar aquí en mi querida UNAM y no menos querida Facultad de Química, participando en este merecido homenaje a un gran académico como lo es el doctor Rafael Castillo Bocanegra. La doctora Alicia Hernández me hizo el honor de invitarme para dar una pequeña reseña de mis recuerdos en la Facultad de Química relacionados con nuestro ilustre homenajeado. La verdad es que creo que mi amiga Alicia me invitó porque soy la memoria más antigua del doctor Castillo en esta facultad. Son muchas y loables palabras que deseo decir sobre el doctor Castillo en este breve espacio de tiempo, para expresar mi admiración académica y debo decir agradecimiento por la amistad que me honro en tener con él. Tuve el privilegio de compartir 43 años de mi vida académica, ¿ya me perdí?, con el doctor Castillo, de manera que mis palabras no son una retórica ocasional, sino la verdad de hechos, sentimientos y emociones, entremezclados con recuerdos de toda mi vida profesional. Por otra parte, la semblanza curricular que se presentó deja en claro por qué se está haciendo este homenaje a un personaje tan destacado en la vida académica de esta universidad, por lo que no haré referencia a la historia del doctor Castillo, sino a mi propia historia y cómo él influyó en ella. No están para saberlo, pero tengo aproximadamente 50 años de conocerlo. Fue en mis últimos semestres como alumna de la carrera de QFB. Él ya se había recibido de la misma carrera y ya daba clases de química orgánica. Y estaba en espera de irse a cursar el doctorado a la Universidad de California. En ese entonces, aún sin conocerlo, yo decidí adelantar una materia de mi plan de estudios para terminar la carrera más pronto. Así, me inscribí para hacer el examen extraordinario de una materia optativa que, de acuerdo a compañeros de generación, estoy tomando la de Alicia, va a conocer mis secretos. Perdón. Me inscribí para hacer el examen extraordinario de una materia optativa que, de acuerdo a mis compañeros de generaciones más adelantadas, era muy fácil, y con aprenderse los apuntes del semestre anterior ya estaba uno del otro lado. Esta decisión la tuvo la mayoría de compañeros de generación y de otras. Normalmente, el examen lo ponía el profesor titular de la materia. Me refiero particularmente a la materia Química Legal 2. Todos ahí ya habíamos cursado Química Legal 1 como obligatoria, pero los ahí presentes no contamos con que el profesor que puso el examen no era el titular, sino el profesor de laboratorio, quien, al enterarse de que se decía que su materia era barco seguro y que la queríamos pasar, como quien dice, al vapor, hizo una modificación, que dado que la materia era teórico, debía, si uno la cursaba, era teórico-práctica, adicionalmente realizaríamos un experimento que significaría el 50% de la calificación final. El día del examen, el salón estaba repleto de estudiantes. Al terminar el mismo, pasamos al laboratorio y repartieron a algunos unos pedazos de ropa con manchas de sangre. La prueba consistía en la determinación del grupo sanguíneo. A otros repartieron vasos con Coca-Cola, a los vasos les adicionaron en nuestra presencia una muestra de un polvo blanquecino que el profesor mezcló con un agitador. La prueba consistía en determinar la presencia de anfetaminas o meprobamato en la Coca-Cola. Y ahí fue cuando el barco se hundió. Nos dieron una semana para realizar el ensayo. Para mi desgracia, a mí me tocó el fatídico vaso y empezó entonces un calvario, ya que no encontraba en la biblioteca algún texto o revista que detallara un análisis ad hoc para la determinación de presencia de anfetaminas o meprobamato en una bebida. Alguien me dijo, en el laboratorio D del cuarto piso del edificio A, aquí viene la historia del doctor, hay un profesor que es un verdadero sabio. Pregunta por el profesor Rafael. Ahí me dirigí y sucedió que se trataba nada menos que de Rafael Castillo. Era un joven profesor que me recibió muy amable y sonrientemente y ofreció ayudarme. Él me facilitó el material y me fue guiando paso a paso y con gran disposición para dilucidar mi problema. Me dijo, vas a detectar cualquiera de los dos compuestos por cromatografía, comparando con 3 estándares. pero yo nunca había hecho una cromatografía en capa fina, porque cuando estudié, esto es en el precámbrico, de verdad, esta técnica no estaba dentro del programa de prácticas de ninguna de las orgánicas. Así que con él corrí mi primera cromatografía en placa en capa fina. Me enseñó a hacer los capilares, las mezclas de disolventes, así como las plaquitas de vidrio en portaobjetos con la gel de sílice. Me recomendó conseguir unos estándares de las sustancias de prueba y mediante una receta médica de un familiar, casi se vendían las benzedrinas sin receta. Compré un frasco de comprimidos de benzedrina, pensando que por descarte sabría si era una u otra sustancia en la muestra problema. Otra situación era que yo aún no había cursado la materia análisis de medicamentos. En esa ocasión, el profesor Castillo, además, me enseñó las bases para extraer un fármaco de un medicamento y, por otra parte, de una bebida. Gracias a su ayuda pude pasar la materia. Hasta la fecha le estoy agradecida, doctor. Posteriormente, el doctor Castillo, como ya bien dijo el doctor Casimiro, se fue varios años a estudiar el doctorado fuera del país. Y cuando regresó para formar parte de la planta académica de la facultad, yo estaba cursando la maestría que en aquel tiempo se llamaba Maestría en Farmacia, entre paréntesis, Síntesis de Fármacos. De hecho, tomé una materia con él y descubrí que era un maestro excepcional. Recuerdo específicamente cuando nos impartió el tema de las complicadas reglas de Woodward-Hoffman y también la clase de estereoquímica. Verdaderamente el profesor hacía arte de sus clases y por otra parte tenía un don para ayudar a comprender conceptos complejos. El doctor Castillo se convirtió en el primer farmacéutico, y creo que el único, y en el futuro también, que ha ocupado la jefatura del Departamento de Química Orgánica. Él tuvo el cargo durante un largo periodo. Yo en esa época daba clase de análisis instrumental y él me invitó, como ya me conocía un poco de esa experiencia que tuvimos, me invitó a impartir clases de química orgánica. Años después, como también vimos en la semblanza presentada por el doctor Casimiro, fue nombrado jefe del departamento de farmacia. Para ese entonces yo ya era parte del personal académico de ese departamento. Como jefe, el doctor Castillo conservaba su carácter alegre, jovial, era muy carismático, optimista y muy activo, pero al mismo tiempo era de temple muy firme y hasta estricto. De hecho, el doctor Castillo fue mi sinodal en mi examen de maestría y en el examen de doctorado, en donde me puso en severos problemas. No se me olvida. Él logró dentro del departamento un ambiente de orden, trabajo y cordialidad entre colegas y siempre estaba dispuesto a ayudar a los demás con algún reactivo, prestar algún equipo o simplemente dando un buen consejo. Más que un maestro, el doctor Castillo se transformaba en un amigo que escuchaba a su colega y brindaba palabras para que éste pudiera avanzar en su trabajo. Además de sus múltiples méritos académicos, él posee una característica polifacética, ya que del mismo modo ayudaba a uno en el trabajo académico, como a despegar una junta esmerilada, o componía la puerta del cubículo o un cajón desvencijado. En una ocasión, recuerdo que acudió a mi cubículo, bueno, no era mío, era de la universidad, pero así decíamos, a mi cubículo, en compañía del doctor Gabriel Navarrete, su entonces estudiante de doctorado, y notó que la puerta estaba colgada. Le expliqué que el personal de mantenimiento no tenía la pieza para arreglarla. El doctor se volvió hacia Gabo y le dijo: Creo que Chabelita necesita de nuestra ayuda. Acto seguido, fueron a su laboratorio y regresaron con la caja de herramientas. Sacaron la puerta de sus goznes, le pusieron calzas, hicieron varios arreglos, la volvieron a ensamblar y colocar en su lugar. Creo que hasta la fecha ha de funcionar bien. Cuando a uno se le atoraba la carreta en el trabajo en el laboratorio, por ejemplo, que se pegaran dos juntas esmeriladas en el rotavapor, había que recurrir a la famosa caja de herramientas del doctor Castillo, de donde salía aquel palo mágico con el que no solo la junta esmerilada se despegaba, sino también cualquier tapa adherida a un frasco. Él es un gran melómano y recuerdo que en una ocasión le platiqué que viajaría a San Francisco, donde él había vivido, y me preguntó que si yo conocía la banda de Pink Floyd. ¿Qué? Le dije. Me contestó, mira, Chabelita, la música clásica no es la única que existe. Debes ampliar tus horizontes musicales y esta es la ocasión. Ya que viajas a los Estados Unidos, cómprate el disco, se llama The Wall, y de paso me traes uno a mí, porque aún no ha llegado a México. Le traje el disco de pasta de 33 revoluciones por minuto. Aún no existían las cintas ni los CDs, todavía era el precámbrico. Así conocí a Pink Floyd a través del doctor Castillo. En este punto quiero decir que pienso que todos los logros del doctor Castillo en su vida profesional se dieron por un talento innato en él, pero definitivamente un elemento y no químico que catalizó su reacción de éxito fue la presencia en su grupo de trabajo de la doctora María Alicia Hernández Campos. No me resta más que decir que muchos académicos hemos dejado la UNAM por retiro, pero estoy segura de que no hay muchos como el doctor Castillo que al irse dejan una huella y un legado muy importante. Ahora que se ha retirado de la academia, doctor Castillo, le deseo lo mejor, hoy y siempre, y estoy segura de que con sus enseñanzas y su dedicación, muchos de sus alumnos han llegado tan lejos y exitosamente en sus vidas. Muchas gracias. Ayúdame,
Pues sí, el doctor Castillo era el carpintero del laboratorio 122. Él construyó las mesas que son aparte de las que tienen los gabinetes. Él construyó varias mesas, la que está al fondo pegada a las ventanas, donde tenemos nuestras herramientas de trabajo con el horno de microondas casero. Entonces ahí la hizo junto con el doctor Francisco Hernández Luis, y también construyó las mesas para las computadoras con ayuda de sus estudiantes de doctorado y maestría y licenciatura en turno, por ejemplo Gabriel. También y Sergio ayudó a construir la mesa que está con el hidrogenador, en donde está su nombre ahí abajo, ¿verdad, Moreno? Está. Aquí estuvo el Moreno, firma, ¿no? Entonces, pues a todos les tocó hacer un poco de química, mucha química, pero también mucha talacha de carpintería, de uso de herramientas, y en el laboratorio todavía va el doctor Mario Figueroa, doctor Alicia, vengo a la caja de herramientas del doctor Castillo, la abre. El doctor Lira, que además tiene su cajita de herramientas, de todas maneras va a buscar herramientas al 122. Y a mis niños en turno, que James, Alan, Joshua, David y Dante y todos los que andan por aquí, ya saben el secreto del palito, ¿verdad? Así es. Cuando no pueden, pues creo que aprendí bien el secreto del palito, pero lo que nunca he aprendido, doctor, es a doblar el vidrio. Como dijo ayer el doctor Marco, no ha habido quien le gane en hacer el uso del fuego y el vidrio para hacer maravillas. Yo creo que en alguna otra época trabajó en la feria de San Miguel Allende o algo así, haciendo figuras. Ok, bueno, continuamos con este coloquio, tertulia o como se diga, en el que sigue la doctora Olivia Soria de la UAM, quien ha sido nuestra amiga, colega, compañera, hija académica también por muchos años. Olivia, bienvenida, gracias.
Buenas tardes a todos y a todas. Es para mí un honor estar aquí en este homenaje, en tan merecido homenaje al doctor Rafael Castillo. Rafael, porque un día se me ocurrió decirle doctor y bueno, casi entró en crisis porque creía que ya me había enojado con él, ¿no? Entonces, bueno, yo titulé esta presentación como maestro, colega y amigo, porque eso es lo que ha sido Rafael a lo largo de todo el tiempo que lo conocí. A ver si estoy... Salió de su querida Villahermosa, Tabasco, para iniciar en 1965 sus estudios de licenciatura en Química Farmacéutica Biológica en la Facultad de Química. Como ya lo dijeron, posteriormente se trasladó a San Francisco, a la Universidad de California en San Francisco, para hacer sus estudios, para realizar sus estudios de doctorado. Lo conocí yo en 1977, cuando yo estudiaba la maestría en síntesis orgánica en la facultad. Recuerdo claramente que en el laboratorio llegó Ada Cázares, la doctora Ada Cázares, muy contenta porque habían regresado de San Francisco dos doctores que además eran sus amigos, dos nuevos doctores que se habían incorporado al departamento de farmacia, el doctor Rafael Castillo y el doctor Carlos Ramón García. A Carlos Ramón yo lo conocía desde la preparatoria, era como nuestro ejemplo a seguir en Tijuana, y sabía de sus sueños de crear una carrera o una facultad de farmacia. Así que yo junto con un grupo de compañeros lo seguimos y nos venimos a estudiar a México para formar parte de ese sueño. Cuando supe que estaban aquí ya ubicados, los fui a visitar para que me platicaran de sus experiencias en el doctorado y me recomendaran algunos libros nuevos de química orgánica, porque en los tiempos en que nosotros estudiábamos no era tan fácil el acceso a la información. La biblioteca pues no tenía tantos libros actuales, teníamos que hacer búsquedas impresionantes en los libros que ahí se encontraban. Recuerdo mi sorpresa al encontrarlos en un cubículo y saber que no tenían laboratorio para trabajar. En ese entonces yo no entendía cómo era que había laboratorios que se utilizaban como oficinas por investigadores que ya no hacían investigación, mientras que investigadores jóvenes con conocimientos de frontera no tenían un lugar donde trabajar. Esa pregunta me la sigo haciendo todavía porque la situación, aunque han pasado más de 40 años, sigue siendo la misma. El doctor Castillo, sin conocerme, me recomendó y prestó varios libros y fue el comienzo de una gran amistad que ha perdurado desde entonces. Quiero aprovechar estos momentos para hablar del doctor Castillo como el gran ser humano que es. El doctor Castillo fue profesor de la Facultad de Química durante más de 45 años, impartiendo cursos de manera ininterrumpida en las diferentes áreas del conocimiento de la química farmacéutica, tanto en licenciatura como en posgrado. Pero lo más importante no fue cuántos cursos impartió, sino la forma de enseñar. Sus clases de química orgánica en la licenciatura de QFB fueron la motivación para que varios de sus estudiantes se involucraran desde temprana edad en las etapas de investigación en esta área del conocimiento. Sus cursos eran muy solicitados porque además de transmitir conocimientos, contagiaba su pasión por la química y por la investigación. Enseñó a sus alumnos a ser disciplinados, a tener rigor científico y a adoptar una ética profesional que lo acompañaría toda la vida. Cuando era invitado a otras universidades como la Universidad de Baja California, Guanajuato, Morelos, a impartir seminarios, siempre se hacía acompañar de un grupo de estudiantes o de alumnos que estaban en su laboratorio, que tenían potencial para ser futuros investigadores, para que impartieran parte de los temas y así se foguearan en los diversos públicos del área. También llevaba a los alumnos desde tempranos inicios en el laboratorio. Les estoy pasando fotografías de las diferentes etapas del doctor, porque una fotografía habla más que 1000 palabras y muchos de ustedes se reconocerán en esas fotografías. Espero que se reconozcan, porque ya pasaron muchos años, ¿no? Entonces, también llevaba a los alumnos desde etapas tempranas de su formación a congresos, a defender sus trabajos y a conocer avances de su campo de conocimiento. Además, no olvido mencionar que a sus alumnos les enseñó que la vida no es solo estudiar química y estar en el laboratorio, sino que es necesario apreciar y disfrutar la belleza de las cosas que lo rodeaban y disfrutar de una buena comida y un buen vino en compañía de los amigos. La fotografía, las plumas fuentes, el cine y la música son sus pasiones y lo compartía con amigos y alumnos. Todavía recuerdo y extraño nuestras sesiones de cine los viernes en la noche, que espero que nadie se haya enterado en aquellos tiempos que teníamos sesiones de cine, porque estábamos calladitos, donde nos pasaba películas que para él habían sido importantísimas y durante toda su juventud. Entonces ahí conocimos actores que nosotros, aunque no estamos tan lejos de edad que él, algunos de nosotros, pero que nunca habíamos visto, sentados en los bancos del laboratorio y poniendo atención a todos sus comentarios y explicaciones. En el mismo tenor, recuerdo las convivencias con sus alumnos y alumnas en las carnes asadas que organizaba cada fin de año con la piñata y el intercambio de regalos, donde nunca nadie se quedó sin un regalo. Otra de las actividades que organizaba para mejorar la convivencia entre los alumnos que trabajaban en el laboratorio eran las excursiones. Les voy a dejar las fotos para que se identifiquen, ¿sí? Éramos todos los del L122 más nuestro invitado o acompañante o hijo de laboratorio, ¿no? Pero en todas las fotos está él, en todas casi está él. No. Bueno, entonces les comentaba que la ida al Estado de México a pescar truchas, para algunos era muy satisfactorio y después no las comíamos, o sea, entonces era muy satisfactorio, para algunos de nosotros era traumático quitarle el arpón a la trucha, ¿no? Que después ya no nos las queríamos comer. Entonces visitábamos estos lugares donde uno puede pescar truchas y después hay quien se las cocine. Les comento que se pescaban tantas que sí nos alcanzaban, y éramos muchos los que asistíamos. Consideraba que la asistencia a los congresos como eventos académicos eran eventos académicos, pero también eran eventos culturales, porque no solo se trataba de ir a escuchar trabajos y asistir a las mejores conferencias, sino que era necesario visitar los sitios históricos y culturales más importantes en la ciudad. Por supuesto, cuando el congreso era en San Francisco, la visita era obligada a la Universidad de California y a comer pizzas en el restaurante donde él iba cuando estudiaba allá, y así como la visita al puerto a comernos una sopa de cangrejo y beber un Irish coffee. Y también asistir a Sausalito a comprarnos un helado, ¿no? Bien, entonces, a lo largo de su vida académica graduó a muchos alumnos y alumnas, 29 de licenciatura, 60 de maestría y 20 de doctorado. Al doctor Castillo lo definiría yo como un formador de talentos, ya que identificaba en cada uno de sus alumnos sus capacidades y habilidades para orientar y guiarlos al máximo en sus potenciales. De su laboratorio han salido reconocidos investigadores que actualmente se encuentran trabajando en diferentes instituciones y laboratorios nacionales e internacionales como cabezas de grupo. A continuación, destaco una muestra breve de algunos de ellos que he tenido el gusto de conocer y me disculpo de antemano con los que no menciono o no pude poner aquí porque o no los conocía o ya la memoria no me da para eso. Entonces, entre sus alumnos más distinguidos, el doctor Francisco Hernández Luis, bueno, los doctores y doctora que son los que voy a mencionar, les voy a decir por el nombre como yo los conozco y como los traté porque está muy largo el apellido. Entonces, el doctor Francisco, la doctora Alicia, el doctor Gabriel, ¿dónde anda Gabriel? José Luis, Sergio, Carlos Alberto, Fabián, que no está con nosotros, está en Colombia, Jaime, que está también fuera de la ciudad y no pudo acompañarnos, pero le manda un fuerte abrazo al doctor. Rubén, Toño, Marco Antonio. ¿No está? Sí, ahí está. Carlos, ¿dónde anda Carlos? Acá está Carlos. Oscar, no sé si esté por aquí. Juan Francisco Cortés, Juana Cecilia Juárez Giordano, Pedro, Remedios, ¿Pero dónde estás, Pedro? Remedios, ¿sí? Que está por aquí, que nos visita desde Tijuana. Y bueno, muchos más, y todos los chicos de licenciatura que ahí se encontraban. El doctor Castillo ha sido siempre un promotor del desarrollo de la investigación y además pionero de la investigación en química farmacéutica en México, con la aplicación de métodos computacionales para el diseño de fármacos. Por la naturaleza interdisciplinaria y la importancia de sus investigaciones, se ha mantenido a lo largo de los años con un financiamiento de fondos de CONACYT. Ha logrado integrar un laboratorio bien equipado para el diseño y síntesis de fármaco. Su acción ha ido más lejos al establecer colaboraciones con científicos de diferentes instituciones nacionales e internacionales. Entre sus colaboradores más destacados se encuentran investigadores reconocidos como la doctora Lilian Yepes, como el doctor Froilán Ibarra Velarde, de la Facultad de Medicina Veterinaria, como el doctor Roberto Cedillo de la Universidad de Yucatán, y a quien tuvimos el gusto de conocer en uno de sus viajes, y el doctor Fry Weber del Queen's University en Belfast. También podemos considerar como uno de los legados más importantes del doctor Castillo la base de datos de moléculas derivadas de benzimidazol, que cuenta al día de hoy con más de 450 moléculas nuevas. Creo que en la mayoría de los alumnos que pasaron por el laboratorio L122, pocos se han salvado de sintetizar algún derivado de benzimidazol. Como resultado de estas destacadas investigaciones, ha publicado 135 artículos en revistas indexadas. Desde sus primeras publicaciones en la revista de la Asociación Farmacéutica Mexicana, empezó a trabajar con derivados de benzimidazol y en sus publicaciones aparecen los nombres de Alicia Hernández Campos y Francisco Hernández Luis, que posteriormente pasarían a formar parte de su gran equipo de trabajo. No puedo dejar de mencionar que la vinculación que ha mantenido a lo largo de los años con empresas públicas y privadas con las que ha colaborado con asesorías y desarrollos tecnológicos. El doctor Rafael Castillo es un científico prolífico, visionario, persistente y comprometido con proyectos siempre enfocados a resolver los grandes problemas nacionales de salud. Y su trayectoria en investigación ha sido reconocida dentro y fuera de la universidad con premios que ya se mencionaron, pero que yo quisiera destacar dos de ellos. El premio que le dio la Asociación de Químicos Farmacéuticos Biológicos, porque son los que tuvimos el honor de acompañarlo y pasar con él un muy buen rato, en compañía de todos los QFBs que lo tienen en gran estima, y de su familia y sus alumnos. Y el premio, el Juchitán, o algo así, Juchitán, que le otorgó el gobierno de Tabasco como un investigador de alto reconocimiento en ciencia y tecnología. También fuimos invitados ahí, lo acompañamos Alicia, Fabián, Jaime y una servidora, y durante nuestra estancia ahí nos llevó a conocer todos los sitios donde había estado en su infancia, a comer las cosas que comía, a beber el pozol, que yo nunca lo había probado y bueno, me atreví, pero como a los 42 grados de temperatura uno toma lo que sea, ¿no? Entonces nos dio el pozol y pues hasta rico me supo a mí. Entonces estuvimos con él acompañándolo y no podía faltar, obviamente, aunque no está en Villahermosa, pero la visita a Palenque, Chiapas, ¿no? Entonces, donde pasamos ratos muy agradables. Le agradezco, acabo de estar hace 3 semanas en Villahermosa, recordando todo lo que habíamos visitado con Rafael, y en Palenque a 42 grados, ¿sí? Igual, no cambia, ahí no cambia la temperatura, ¿sí? Entonces, no puedo dejar de mencionar, como dice, ya me perdí yo también. Por último, quiero resumir y describir al doctor Rafael Castillo Bocanegra como un excelente maestro, un investigador destacado, generoso como ser humano, buen corazón y un sentido del humor que no se acaba. El amigo que siempre está cuando lo necesitas. Demos de pie un fuerte aplauso al doctor Rafael Castillo Bocanegra, una persona excepcional del que todos hemos aprendido algo y estamos orgullosos de que nos haya compartido sus conocimientos y distinguido con su amistad. Gracias, doctor Castillo. Gracias, Doc. Gracias, Rafael.
Muchas gracias, doctora Olivia. Amiga, compañera de cuarto, cuando viajábamos a los congresos, etcétera. Continuamos con otra gran compañera y amiga, colaboradora por muchísimos años, la doctora Lilian Yepes del Instituto Mexicano del Seguro Social, con quien hemos también tenido una relación importante, y el doctor Castillo ha sido con ella la mancuerna en la parte biológica y química de un gran número de publicaciones que a estos dos doctores les ha permitido estar, y a mí también, estar en las primas de desempeño, en el trabajo, los estímulos, el SNI, todo. Y muchas gracias por estar aquí, Olivia. Olivia sí, pero Lilian aquí está.
Sí, ahorita vamos a poner una pequeña presentación que estuve elaborando por unos buenos días. No encontraba suficientes fotografías para poderles mostrar lo que es el doctor Castillo, el doc, con mucho cariño. Sí, ¿me la pones? Bien. Bueno, pues primero que nada les agradezco a todos los organizadores de este evento Para mí es un honor estar aquí para hablar del tan querido doctor Castillo. ¿Sí? Entonces, todos lo conocemos como un profesor, como acaba de resumir esta Olivia, ¿verdad? Con grandes méritos, todo un académico, todo un científico y todo un gran amigo, todo un ser humano. Yo les voy a platicar un poquito la manera en que inicié esta colaboración con el doctor Castillo y su grupo de investigación. Esto fue de manera indirecta porque en alguna ocasión aquí en la Facultad de Química invitaron a dar una plática al doctor Cedillo, Roberto Cedillo, que ahí se los señalé. El doctor Cedillo vino a platicar del efecto del albendazol sobre la yarda intestinalis. Después de esta plática se acercó el doctor Castillo, platicó con el doctor Cedillo y le comentó, lo llevó a su laboratorio y le comentó que tenía muchos frasquitos llenos de derivados benzimidazólicos, pero que requerían una evaluación biológica contra parásitos protozoarios y también helmintos. Entonces, de regreso al centro médico, el doctor Cedillo me comentó y me dice, yo en ese entonces estaba trabajando con triquinela, estaba trabajando cuestiones inmunes, y me comentó el doctor Cedillo, oye, fíjate que pasó esto, el doctor Rafael Castillo quiere que comencemos, él iba a hacer toda la parte de evaluación con protozoarios, y yo iba a entrar con triquinela a la parte de helmintos. Entonces, yo al principio no estaba muy segura de querer participar en esta colaboración. La razón era de que nunca había entrado a esta dinámica de evaluar compuestos y ver su actividad y su efecto, su mecanismo. Entonces, al principio me resistí, pero pues me hicieron manita de puerquito, era mi jefe, y entonces comencé y así inicié la colaboración con el doctor Castillo. Aquí es triquinela y entonces comenzamos a evaluar en un modelo murino el efecto de varios compuestos de los que el doctor Castillo tenía en esos frasquitos y evaluábamos la actividad de estos compuestos a nivel enteral y sistémico y veíamos finalmente cuantificamos la reducción de la carga parasitaria. Pero poquito a poquito también nos comenzamos a adentrar a investigar un poquito lo que era el mecanismo de acción, que es un poquito lo que les muestro abajo. Pero bueno, esto fue con un primer compuesto, que fue el GNB14, que aquí está el autor, Gabriel, y tuvimos mucho éxito con este compuesto. Pero me estoy saltando algo, muy al principio nos dio unos compuestos el doctor en sus frasquitos, eran 16 frasquitos, todos rotulados, y tenían una anotación que era RSD-1, 2, 3, 4, 5, hasta el 16. Entonces, en ese momento estaba un estudiante de licenciatura que había contactado al doctor Francisco Hernández y me lo llevó. Él era químico de la FE Zaragoza. Y entonces dije, bueno, pues es el momento en que comencemos a evaluar algo y demás, ¿no? Pero nos torturábamos tratando de entender qué compuesto era. RSD. Pues no entendíamos, decíamos, no, es que no sé qué estructura es, pues él dice que es un benzimidazol. Bueno, entonces pedimos una cita para que nos iluminara y pues nos iluminó. Resulta que era la clave de su estudiante, era Remedio Sánchez. Entonces, bueno, de ahí aprendimos que todos los que nos iban a llegar tenían esa nomenclatura, ¿sí? Así como el GNB14. Ya, después de eso ya no tratamos de investigar absolutamente nada, los evaluábamos. Entonces, bueno, empezamos con la triquinela. A raíz de esta colaboración que inicié, realmente yo nunca pensé que fuera a ser un área tan interesante y tan productiva. Realmente me comenzó a enseñar muchas cosas y pues fue muy bonito aprenderlas. Participé con él, ya estando en esta colaboración, en varios comités tutorales y exámenes de grado, que aquí les muestro algunos de ellos. También, así como dice Olivia, se pueden reconocer cuando eran jóvenes y bellos. Hay varios. Recibimos algún premio de Glaxo con algunos de los compuestos que evaluamos y pues en estas asesorías, en esas colaboraciones en las que yo participaba con él y con su grupo, pues estaba en los exámenes de grado y yo soy bióloga, no soy química. Entonces, pues yo me quedaba en la parte biológica, en la parte de asesoría biológica, de los ensayos biológicos y todo esto. Entonces, cuando participaba en los exámenes con el doctor y esto, yo siempre me quedaba en la parte biológica, ¿sí? Y entonces alguna vez en algún comentario que el doctor Francisco Hernández nos hizo saber, alguien, no estoy segura si era sinodal o era parte de la audiencia, comentó, esta química sabe mucha biología. Entonces, y de ahí me quedé con eso, una química que sabía mucha biología. Y bueno, pues así pasamos. Hicimos muchos proyectos, elaboramos muchos proyectos de investigación con su grupo. En ese momento vitales eran la doctora Alicia Hernández, el doctor Francisco Hernández, eran los ejes junto con el doctor Castillo. Y elaboramos muchos proyectos de investigación. Nuestro punto de reunión era o la sala de juntas del edificio E de la Facultad de Química o en el famoso laboratorio L122. Ahí pasábamos días, muchos proyectos los elaboramos y tardábamos en elaborarlos, poníamos el cañón, veíamos, discutíamos, hacíamos, opinábamos y bueno, nos llegaba el momento en que teníamos que subir a la plataforma de CONACYT esos proyectos para obtener apoyo y llegaba el día límite y nosotros todavía seguíamos laborándolo. Entonces empezamos con ansiedad y todo esto, y el doctor Francisco Hernández, para animarnos, decía, el tiempo se acaba hasta el último minuto. A las 11:59 de este día todavía los podemos subir. Y eso nos animábamos, porque eran las 7, las 8 de la noche y no podíamos subir los proyectos a la plataforma, pero lo lográbamos finalmente, con la participación muy importante de muchos de los estudiantes del doctor Castillo, ¿verdad? Pedro fue uno de los que nos ayudó mucho en algunos proyectos más difíciles que tuvimos. Bien. Aquí también, así como mencionan que el doctor Castillo construyó mesitas y todo, construyó un refugio atrás de un refrigerador que todos usábamos, ¿sí? Y ahí teníamos a una colaboradora pequeña, que era mi hija, que tenía como 9 años, y que pues trabajó duro con nosotros también, porque nos llevábamos días enteros, a veces nos quedamos la noche trabajando. Entonces ella era su lugar donde se dormía, ¿sí? Después descubrí que era de varios, el lugar donde se dormían, pero bueno, entonces, vamos a ver. Elaboramos todos estos proyectos, obtuvimos financiamiento varios de grupo, individuales, por CONACYT, también de GAPA, y comenzamos a ver otros compuestos, otra vez el CMC20, otra clave, ¿verdad? Aquí está el autor, pues Carlos Méndez Cuesta. Entonces ese es CMC20. Entonces, de estar viendo qué tanto se reducía la carga parasitaria, si no se sentían mal los animalitos, etcétera, pasamos a los mecanismos. Y en este caso, comencé a trabajar ya con Giardia intestinales. Y aquí lo que les muestro es el efecto de este compuesto. Y ustedes pueden ver el efecto tan tremendo que tiene este compuesto. Uno de los compuestos con los que tuvimos más éxito, ¿sí? Y aquí el autor de todo este trabajo también está aquí, que es el doctor Félix Matadamas. Bueno, el compuesto Carlos, y todo este microscopio y todo se obtuvo en la tesis de doctorado de Félix Matadamas. También estuvimos trabajando mucho el mecanismo, iniciamos análisis proteómico, biología molecular, o sea, comenzamos a diversificarnos para ver el efecto de estos compuestos. Y trabajamos con leishmania, aquí podemos ver el efecto, ustedes pueden ver de una manera sorprendente cómo lo lisa, destroza la membrana celular, ¿sí? Y así seguimos. Todos estos resultados que obtuvimos de estos proyectos, pues llegaron, se finalizaron su reporte en artículos y se publicaron varios, estos no son todos los que se sacaron de todo eso que estuvimos haciendo en revistas de difusión internacional. Realmente tuvimos un gran avance en el conocimiento de la estructura del benzimidazol y su actividad parasitaria, porque ya fuera por diseño racional, aprendí eso del diseño racional, o por modelado molecular, también lo aprendí ahí, y aprendí 2 que 3 cositas. Alicia se empeñaba mucho en enseñarme lo de sus carboxamidas, que si la 1, que si la 2, soy bióloga. Entonces no sé todavía no le hallo a eso, ¿no? Bueno, quiero mencionarles en especial un proyecto de grupo, porque además teníamos un grupo multidisciplinario, porque no nada más éramos el doctor y yo, y su grupo importante con el doctor Francisco y con Alicia. También estaban investigadores del Simposio, de la Escuela Nacional de Ciencias Biológicas, de aquí de la Facultad de Medicina, e interactuábamos mucho, muy bien, nos llevábamos muy bien, también por reglas que siempre se obedecieron y mucho por la actitud y por la calidez del doctor Castillo. Entonces, en el 2012, el doctor recibió un correo por parte de CONACYT donde le avisaban que el Fondo Sectorial de Investigación para la Educación, CEP-CONACYT, antes eran CEP-CONACYT, realizaría un congreso que se ha denominado Congreso Nacional de Ciencia Básica 2012, Casos de Éxito. Y escogieron en ese entonces este proyecto con registro 80093 en el área de ciencia básica, en modalidad de grupo, y quien era el doctor Castillo era el responsable técnico, y lo llamaron Casos de Éxito. Fuimos a presentarlo, esto fue ahí no me acuerdo si fue en Cancún, y pues aquí nada más les resumo por qué lo nombraron Casos de Éxito. Este fue un proyecto de 3 años, y aquí les resumo las publicaciones que salieron de ese proyecto, los recursos humanos, tanto de licenciatura como de posgrado, y reconocimientos a estos proyectos. Entonces fue un caso realmente de éxito. Entonces, esta colaboración que mantuve con el doctor Castillo fue por más de 25 años, ¿sí? 25 años que pues realmente fueron de gran conocimiento, de gran alegría, siempre salía la sonrisa, sobre todo de Alicia, ¿verdad? Siempre la carcajada que nos sacaba, y pues lo disfrutamos mucho, y fueron cosas muy interesantes. Bien, pensé que se habían desaparecido, pero bueno, como ya se ha mencionado mucho, algo característico del doctor Castillo, del doc, es que no nada más, él no es un adicto al trabajo para nada, o sea, él es muy trabajador, él trabaja, sabe hacia dónde, cómo, pero nunca se olvida de disfrutar. Trabajábamos, les comentaba de estos proyectos, o en reuniones para presentar avances de los proyectos. Imperdonable la salida a comer, ¿sí? A la fonda italiana aquí en San Jerónimo o algún otro lado, íbamos a comer. Siempre disfrutó mucho de convivir con sus estudiantes, con sus colaboradores, pero también con la familia de estos. Entonces aquí tenemos, ahí mencionan del lado superior izquierdo, una visita del doctor Ismael Hidalgo. Doctor Ismael Hidalgo era un experto en las células CACO2. Las CACO2, cuando nosotros estábamos haciendo un proyecto, se envió con apoyo de CONACYT a un estudiante, a Raúl Morales, a trabajar por un mes, no recuerdo, un mes, 3 meses, allá con el doctor Ismael Hidalgo. Y de ahí aprendimos a comenzar a calcular la permeabilidad aparente, ¿sí? Entonces era un gran hallazgo. Al doctor se le invitó para venir aquí a dar unas pláticas sobre CACO2, cómo se cultivaba, etcétera, etcétera, porque no es cualquier cosa, como ustedes sabrán. Y pues el doctor y la familia de Ismael Hidalgo, que está ahí su esposa, sus hijos, y la familia del doctor Cedillo fueron a una visita a Teotihuacán. Entonces el doctor siempre era una persona que le gustaba tener esta relación de amistad con todos los colaboradores, ¿sí? Abajo tenemos, bueno, en el centro tengo una relación muy especial que tuvo el doctor Castillo con el doctor Cedillo. Se entendían muy bien los dos, ¿sí? Y pues idas a Metepec o a Yucatán con el doctor Cedillo, que ya está trabajando allá. También, pues ya lo mencionaron, pero íbamos mucho, bueno, yo no tuve la oportunidad de ir tantas veces, pero al doctor le fascinaba ir a Sultepec, a la pesca de truchas, que ya mencionó Olivia, ¿verdad? Que era todo un evento, sacábamos truchas enormes y nos las preparaban y nos las comíamos. Recuerdo mucho en alguna ida, precisamente en la que está en la foto, la impresión ahí en la foto en el centro, de esta Olivia llevando toppers chiquitos, medianos, grandotes y más grandotes. Y finalmente era la que podía ayudarnos a colocar a todas estas truchas, ¿no? Pero bueno, eran todos unos eventos muy bonitos, de mucha convivencia. Ahora, Tuvimos mucha interacción, mucha asistencia y participación en congresos nacionales e internacionales. Como ya mencionaron, el doctor Castillo se preocupaba porque sus estudiantes fueran, ¿sí? Que se foguearan en estos eventos, que participaran, que platicaran con investigadores de mucho nivel, y pues así fuimos a congresos de la Sociedad Americana de Química, ¿verdad? Y ahí fuimos. Quiero mencionarles, ahí va a salir de aquí en adelante, perdón, nuestra colaboradora más pequeña, la que se dormía entre el refrigerador y la ventana, que está a mano izquierda. Ella era la más entusiasta en estos congresos porque siempre estaba atenta a cuál nos tocaba el año que entra. Entonces, estaba ahí, también estaba presente en los pósters. Hay otra fotografía donde la pobre está sentada en una silla alta, recargada en el póster, dormida en Orlando, ¿no? Pero bueno, participamos en todos esos eventos, pero una vez que cumplíamos con nuestra actividad académica, venía la parte de convivencia muy estrecha, que era visitar esos lugares donde nosotros íbamos. Y pues aquí les presento en Orlando, al doctor casi se lo devora un dinosaurio, ¿verdad? Ahí está en el centro, fuimos en Orlando y pues siempre llevábamos. En esa ocasión fuimos a Jurassic Park, nos subimos en una lanchita, nos devoró el dinosaurio y comenzamos a subir. Iba la maestra Inés, que no sé si esté por aquí, pero bueno, nos subimos. Y en ese momento que estábamos sube, nos dijo Francisco Hernández, nos dijo, agárrense fuerte, porque recuerden que todo lo que sube baja. Pues lo acababa de decir cuando nos bajaron por la cola del dinosaurio y fue un splash. A mí se me zafaron los lentes, mi hija casi quedó en el asiento de adelante, o sea, fue impresionante, pero nos divertíamos mucho de esa tensión que llevábamos también en preparar y presentar los pósters. La ida a San Francisco, que se ha mencionado mucho, creo que esta es icónica. Ahí estamos toda la legión que acompañaba al doctor Castillo a estas salidas, ¿sí? Están arriba en San Francisco. Están aquí, pues está el doctor Francisco Hernández, Blanca, su esposa, el doctor Cerillo, el doctor Castillo, su servidora y la colaboradora pequeña. Entonces estamos así. En Alcatraz, y en San Francisco, pues conocí a Rubens, no podía dejar de conocer Rubens, entonces lo conocí, ¿verdad? Y fuimos a Alcatraz, y el doctor siempre con su buen sentido del humor, ¿sí? Tomando las fotos en momentos especiales y después nos las daba, gozaba mucho con eso el doctor. Ahí está también nuestra colaboradora atrás de las rejas, y pues nos divertíamos mucho. Quiero mencionarles, no tuve fotos lamentablemente para platicarles algo que nos pasó en Sausalito. Fuimos al Pier 41 porque queríamos ir a Sausalito. Tomamos el ferry, íbamos muy contentos, vimos el Golden Gate, todo padrísimo y demás. Y en determinado momento el doctor Cerillo, el doctor Castillo y Alicia se fueron a lo alto del ferry, porque el doctor llevaba su cámara, ya saben, y compartía con el doctor Cerillo ese hobby y aparte el gusto por la música, ¿sí? Entonces se platicaban mucho y todo. Entonces subieron y nos quedamos abajo, lo que era Francisco, Blanca, mi hija y yo. Llegamos al muelle de Sausalito, ¿sí? Nosotros nos bajamos y pensamos que nos seguían ellos. Pues no, bajamos el muelle, caminamos y nosotros volteábamos y ellos seguían tomando fotos, viendo a todos lados y nunca se dieron cuenta que el ferry se alejó del muelle. Entonces, lo único que hicimos fue bye bye, bye bye, y decíamos, ¿y ahora qué? Pues vámonos a comprar el helado y a conocer Sausalito, ¿verdad? Ellos se tuvieron que regresar a Pier 41 y horas después regresaron a Sausalito. Así eran ellos, se posesionaban y todo, ¿no? Pero era muy, muy divertido con ellos. Hubo congresos nacionales como el de parasitología. El doctor, con su inteligencia, con su capacidad, llegó a ser realmente un gran parasitólogo. A veces lo invitaban más a él que a mí. Entonces, era la estrella. Y había en alguna ocasión organizadoras del evento que las cautivaba el doctor Castillo. que sí las cautivaba con su sonrisa, con su calidez y así su sonrisita. Y bueno, doctor Castillo para acá, doctor Castillo para allá, y la doctora llévese en cuenta, ¿no? Va ahí siguiendo al doctor Castillo. Pero bueno, en uno de esos congresos fue donde el doctor Castillo, Alicia, conocieron al doctor Fairweather. Del lado derecho, el doctor güerito alto, es el doctor Fairweather. También está el doctor Javier Ambrosio, con el que participaba en muchos eventos, en muchos congresos, en proyectos de la Facultad de Medicina. Y pues abajo en Guanajuato. En uno de estos eventos en Guanajuato, el doctor, uno de parasitología, iba a tener una presentación. Y una noche previa nos pidió a Alicia y a mí que revisáramos su presentación. Y él ya se fue a descansar. Y pues nosotros prestas, ¿no? Aquí las expertasas comenzamos a ver su presentación. El doctor tenía de 15 a 20 minutos para dar su presentación. Y entonces comenzamos a ver que había, para nuestro parecer, diapositivas cargadas de información. Y decíamos, no, el doctor se va a parar mucho en esto para explicar y demás. Y entonces aquí las expertasas dijimos, quitamos esta, quitamos la otra y se las quitamos. Y le redujimos, según nosotros, la presentación. Al día siguiente que el doctor Castillo presentó, pues se quedó asombrado, así sacado de onda, y nosotros también nos quedamos, le sobraron 5 minutos de su presentación. Entonces cuando vimos eso, pues nada más nos mirábamos y ya, pero el doctor con su experiencia pudo salir al paso, habló algún chistecito además, y ya, le aplaudieron al doctor como era de costumbre, ¿no? Entonces ya después nosotros, Alicia y yo, le confesamos que le habíamos reducido su presentación. Pensamos que se iban a molestar, pero no, el resultado fue una carcajada de todos y seguimos siendo amigos, ¿sí? Pasamos adelante. Entonces esta presentación lo que yo quise es mostrarles esa parte académica que fue exitosa, muy productiva, muy estrecha, pero también es otra parte humana, es otra parte que era característica del doctor Castillo, toda esa calidez que tenía. Él siempre, pues de alguna manera mostró mucho cariño a mi hija y que siempre se lo he de agradecer. Y pues finalmente quiero decir que si yo pudiera resumir la colaboración con el doctor Castillo, diría que fue una experiencia académica y de convivencia inigualable, la más entrañable y completa de las colaboraciones que he tenido en mi vida académica. De esta colaboración nació una amistad estrecha que ha perdurado en el tiempo y que el doctor Castillo, con su sencillez, con su buen humor, calidez, le dio un toque muy especial que siempre va a quedar en mi memoria y en mi corazón. Doctor Castillo, le hago llegar todo mi cariño, mi admiración y reconocimiento por toda su trayectoria. Muchas gracias, doctor.
Muchas gracias, doctora Lilian. Bueno, pues continuamos ahora con la doctora Raquel Mata, una gran investigadora de nuestro departamento, que también a lo largo de muchos años ha colaborado en las cuestiones académicas del posgrado y todo eso, teniendo pues mucho aporte de su parte y su conocimiento en el manejo de los proyectos, en el manejo de los estudiantes y sobre todo la calidad humana tanto de la doctora como del doctor Castillo. Doctora Raquel, por favor.
Ok.
Hola, Doc, ¿cómo está? Como todos les decimos. ¿Bien? Muy bien y encantado, ¿verdad? Mire, doctor, ya le han dicho de todo, pero no le han dicho lo guapo que es usted, lo amable que siempre fue con todos los alumnos, ¿verdad? No solamente los suyos, sino con los de los demás compañeros, ¿verdad? De trabajo. Fíjense que hasta un matrimonio hicimos, ¿se acuerdan? El de Daniel y Remedio, aquí está. ¿Se acuerda que hicimos ese matrimonio, lo arreglamos? ¿Y que se acuerda, doctor, de todas las conspiraciones que hicimos contra las autoridades? Y fíjense, todavía las autoridades no entienden de qué se trata el departamento de farmacia. ¿Cuántas veces, doctor? Digan, ¿recuerda o no? Muy rápido. Muchas, ¿verdad?
Ya,
Muchas veces, directores, coordinadores de posgrado, bueno, de todo, ¿verdad, doctor? Conspirando, pero además con premeditación, alevosía y maldad en muchas ocasiones. ¿Se acuerdan? Bueno, algo logramos. Y ahora no se preocupe, porque ahora queda Rodrigo Aguayo, que me impidió leer. Yo tenía algo preparado, iba a leer, pero dijo que no, que yo no podía leer, que iba a leer en 5 minutos. Entonces, bueno, gracias Rodrigo, porque así improvisé mejor. Entonces a Rodrigo le pasamos la estafeta y él es el encargado ahora de hacer entender qué hacemos en el departamento. Creo que su paso fue fundamental, ¿verdad? Por el departamento, todos hemos contribuido con algo y creo que su paso fue fundamental para el florecimiento pues, de nuestro departamento y sobre todo, doctor, lo más difícil, hacer entender, ¿verdad?, de qué se trata. Hace poco lo extrañé porque fui con Francisco a la misma comitiva, parece que ya me lo supiera de memoria, fíjense. Y seguiremos, pero ahora ya yo decidí que la estafeta la va a tener aquel jovencito que está allá atrás. Doctor, muchas gracias por todo. Usted me recibió, me abrió las puertas. El año que viene van a ser 40 años.
¿40?
Sí. Usted me abrió las puertas del departamento. ¿Se acuerda? Usted me abrió las puertas, me recibió con mucha calidez, yo estaba con mucho miedo, porque imagínese entrar a ser profesora de la Facultad de Química. Y bueno, pero usted me facilitó todo, no la caja de herramientas, sino otras cosas que fueron muy importantes y mi gratitud eterna por eso, doctor. Además creo que caminamos muy bien, hicimos muchas cosas, trabajamos, publicamos juntos, bueno, toda una vida, ¿verdad? Y eso, pues, usted siempre estará presente y recuerde que el mejor líder es el que, sin estar presente, siempre está presente. Y eso es usted. Y ojalá que todos estos organizadores de estos eventos dejen para siempre el nombre del doctor Rafael Castillo para este evento. ¿Sí? Bueno, un abrazo, te doy. Yo también. A pesar de
para usted. Es un recuerdo.
Seguimos, ya casi terminamos, pero gracias, doctora. Y gracias por esa sugerencia que ya yo la tenía en mi mente, yo creo que muchos. A lo mejor a partir de este 10º simposio, este simposio se llamará Rafael Castillo, porque quiero decir algo muy pequeño. Un chico ayer me pasó a mi celular una cartita que le quería dedicar al doctor Castillo, unas palabras. Por el tiempo, Dante, se la voy a leer en petit comité, en privado, pero quiero decir algo muy bonito que dijiste. Después de haber, dice Dante, que anda por aquí, después de haber incursionado por los otros grupos de química orgánica, etcétera, pues él se cambió a la carrera de QFB porque estaba en química. Y dice que aquí dice, entre otras palabras, decidí ser QFB y continuar en la carrera por inspiración y motivación del doc. Me llena de orgullo el haber compartido estas anécdotas del boxeo, porque platicaban de box y también platicaban de lo difícil que son las purificaciones de esos compuestos necios de color amarillo, que cuando manché mi bata de dicho color, me dijo, bienvenido al 122. Aquel que fuera al 122 y no manchara la bata de amarillo, no pertenecía a la familia. Muchas gracias, Doc. Su legado continúa con todos sus alumnos y seguirá por generaciones con sus nietos académicos, porque como se ha dicho, química farmacéutica es sinónimo de Rafael Castillo en México. Gracias, Dante, por tus palabras. Bueno, ¿ya? Sí. Entonces, ya le pongo un pucho. ¿Dónde le pucho? Okay. Sí. Bueno, ya se ha dicho que el doctor Castillo hace 80 años que nació en Villahermosa, Tabasco, y aquí les presento, como él mismo nos enseñó, oye, esta cosa no sirve. A ver, no pinta, digo, no, esta pluma no tiene tinta. Bueno, ahí está la laguna de las ilusiones donde nos contaba el doctor Castillo, y ahorita los veo a ustedes como cuando en las familias los nietos y los hijos pequeños están alrededor de la mesa oyendo las anécdotas de sus padres cuando eran niños, ¿no? Algo así. Entonces, aquí les cuento que el doctor Castillo en la gráfica, digo, en la diapo, no sirve, ¿y en mi control? Bueno, aquí les muestro, ni siquiera se ve el cursor, pero bueno, es la laguna de las ilusiones, donde el doctor Castillo nos dijo que dio su primer beso. También en este lugar, sus amigos, cuando lo acompañamos al Juchimán de Plata, nos contaron que pues ahí se hicieron hombres. Aquí se encuentra, Doc, mire, su mamá, doña Trina, que cuando de pronto hacía yo un berrinche en el laboratorio, en lugar de decirme, Alicia, contrólate, me decía, doña Trina, contrólate. Yo creo que su mamá era un poco exaltada, como yo comprenderé. Ahí está su mami y sus papás. Y el doctor Castillo, joven y guapo. Se formó en la prepa, allá en Tabasco, y desde entonces él se dio a la tarea de enseñar, y nos cuenta también que él desde entonces daba clases en un laboratorio de química y era el preparador de las prácticas de laboratorio. Hueso, Emico, Pochitoque, Nacajuca, Juchimán, Ujat, es la porra de donde vienen los nombres de las computadoras, las máquinas de estaciones de trabajo de nuestro laboratorio. Ay, me voy, sigo. Bueno, se volteó otra vez, por segunda o tercera ocasión, esa foto se ha volteado de cabeza, pero estudió la carrera de QFB en los tiempos de estos doctores, el doctor Madrazo y el doctor Herrán. Aquí está su tesis de licenciatura, ese librito azul verde que está ahí en las tesis en el laboratorio, pues es la tesis del doctor Castillo, la hizo en productos naturales. Y le dedicó la tesis a el teniente de sanidad militar, José, ¿qué? No le alcanzo a leer, Arias. Él es el que lo enseñó en el área o lo enamoró de la parasitología. Aquí en la facultad lo guiaron en su tesis de licenciatura la doctora Espejo y el doctor Giral. Aquí está su título. No sé si no daban menciones honoríficas en ese tiempo, pero no le dieron mención honorífica. Yo creo que tenemos que ir a romper los vidrios de rectoría, como dijera Rodrigo en algún momento. San Francisco fue su cuna de trabajo, del posgrado, se enamoró de San Francisco y ahí tuvo su primera hija en San Francisco, y cada que había un congreso en ese lugar íbamos, porque teníamos que recorrer inclusive los mismos lugares, porque era una remembranza muy importante. Ahí nació su hija, en ese hospital. Su título de doctor en filosofía. Aquí su grupo de trabajo, el doctor, ay, espérenme, espérenme, aquí, su amigo el hindú, Amrit, aquí abajo, doc, es que no me sirve o no lo sé usar, perdónenme, así me pasa en clase, no se preocupen. El hindú, Amrit, aquí está, y el chino, aquí está. No me acuerdo su nombre, pero decía que tenía un compañero muy bueno en síntesis, o era su amigo ahí. Y el doc ahí, siempre de galán, posando. Pascuale es la pizzería que algún día cuando vayan a San Francisco tendrán que buscar, como nosotros pasamos 2 horas buscándola, para poder ir a conocer el lugar en donde dice el doctor que lloraban cuando les iba mal en los exámenes y se echaban su buena pizza de buen tamaño y sus yardas de cerveza. Los artículos que publicó durante su estancia en Estados Unidos, todo que ver con la escualeno sintetasa, sus tutores, su comité tutoral evaluador, aquí está el doctor Kitchman Castagnoli, que hacía metabolismo, que el doctor Francisco y yo conocimos en un curso que tomamos en Drew, el Manfred Walt y Eugene Jorgensen, que pensábamos que iba a venir, pero era otro. Bueno, aquí están sus evaluadores del doctorado, su jurado, que le dio, según nos cuenta el doctor, el pase positivo en 30 minutos. Entonces, que nunca había habido un examen de doctorado tan rápido. Se instaló en la Facultad de Química desde el 70, como profesor, dando clases, siempre con una sonrisa, siempre ayudando, siempre posesionándose de una mesa de laboratorio, porque parecía el alumno Castillo muchas veces. Se agarraba su charola y hacía la práctica y demostraba a los chicos cómo hacerlo. Bueno, aquí hay algunas tesis importantes que tienen que ver. Aquí está mi tesis de maestría, por ejemplo, la del doctor Francisco, muchas tesis de licenciatura, muchas tesis de maestría, de doctorado. Pues aquí estamos en el aniversario del 50 aniversario del posgrado en Ciencias Químicas, pues muy contentos. Sus alumnos se han repartido a lo largo del país y también del mundo. Hay en todos lados, Canadá, Colombia, andan en Europa, en México, en Yucatán, en Campeche, en Oaxaca, Tijuana, etc. Colaboraciones muchas con gente querida de nuestra facultad, como la doctora Helgi, que no pudo venir, pero manda muchos abrazos. La doctora Marina Binaud de Brasil, que estuvo en el simposio pasado aquí, por Zoom. La doctora Laura Domínguez, que se quedó con Rodrigo porque el doctor Castillo se lo entregó y le dijo, yo no puedo con este niño, te lo doy porque tú vas a saber qué hacer con él. Y no porque no lo quisiéramos, sino porque no podíamos enseñarle lo que él necesitaba. El doctor Froilán Ibarra y la doctora, perdónenme, el doctor Froilán Ibarra y la doctora Vera de veterinaria, con quien hemos colaborado mucho tiempo también en el área veterinaria con nuestro famoso compuesto alfa y ahora con el famoso fosfatriclaven. Los doctores de la Universidad de Durango, con los que también tenemos trabajo en cuestiones de ensayos enzimáticos y hemos tenido alumnos en nuestro laboratorio también. El doctor Cedillo, que les presentó la doctora Yepes hace un rato, con quien compartían los calcetines cuando a alguno de ellos se le olvidaba en la maleta. Bueno, el doctor Fairweather, que cuando llegó a México al Congreso de Parasitología, no quería saber nada del Congreso de Parasitología, quería conocer a Rafael Castillo. Entonces entró un día a la sala, recuerdo, y entonces dijo, ¿quién es? ¿Quién es? Dijo el doctor Fairweather. ¿Dónde está Rafael Castillo? Lo quiero conocer. Y entonces me vio al lado y dijo, bueno, también Alicia. Ok, bueno, muy bien. Fue de los pioneros en hacer química computacional, con su famoso artículo del índice de conectividad molecular, y desde entonces él le gusta también la parte del diseño, utilizando cosas físico-químicas, estructura, conectividades y las computadoras. Los benzimidazoles nos han perseguido desde siempre y hemos hecho muchas cosas, y digo hemos porque, como lo han dicho mis amigas que pasaron, pues me ha tocado trabajar muchísimo en el laboratorio con estas moléculas. Entran los análisis por docking, computadora, y yo le dije a Rodrigo un día que me hizo un berrinche, y le dije, tranquilo Rodrigo, la beta tubulina te va a dar de comer. Es cierto, está comiendo de eso. Muchas moléculas, una gran biblioteca de compuestos, con diferentes estructuras, diferentes actividades. El benzimidazol, como molécula privilegiada, pues nos da las bondades de, como un arbolito de Navidad, cuando le colgamos esferas de un color o le ponemos moños de otros, podemos modificar su actividad biológica. Y eso lo aprendimos de Rafael Castillo. Ya mero, aquí terminamos algunas cosas. Publicaciones varias, muchas, árbitro en muchas revistas, evaluador de proyectos con ACID, ahora con ACID con H, revisión técnica de algunos libros también, proyectos y congresos nacionales, internacionales, participación en un montón de seminarios, mesas redondas, etcétera. Ahí hay unos números que ya no casan con la realidad, pero bueno. Y su famoso compuesto alfa, que sin usar muchas herramientas más que su cerebro, y como en el grupo conocemos su diseño in cerebrum, logró tener este compuesto tan activo que sigue siendo el que nos sigue dando de comer. Bueno, aquí está cómo se diseñó a partir del propranolol y el isoproterenol. Sacamos al alfa, y sacamos porque yo, como decía Marco ayer, me quemé mucho las manos con estas reacciones. Muchos estudios que se han reportado al respecto y nos ha dado muchas publicaciones esta molécula. También, como era una roca, pues lo disolvimos haciendo su sal, y pues de ahí también tenemos varias patentes y estamos en pláticas con algunas empresas. Toño, feliz y contento también, creo que me ganaba en las carcajadas. Y pues el doctor aquí poniéndose la bata para que los píxeles no los dañaran. Fuimos a muchos congresos, hemos ido a muchos congresos, y aquí estamos divertidos, también dando cursos. Aquí estamos en Guanajuato, aquí estamos en Boston, en el que estábamos juntándonos para hacer una foto, y nos dijo una persona de la prensa, y nos dijo, yo les tomo la foto con la condición de que una de ellas sea para mi revista, y salimos en el Chemical and Engineering News. Premios, muchos también, como con el Alfa, muchos premios, el premio Canifarma, el Martín de la Cruz, el Juchimán, el del patentamiento, el de excelencia académica, el de docencia en ciencias naturales, premio Universidad Nacional 2018, y cada vez que yo puedo lo presumo por él. Todos los niveles más altos en los estímulos, en el CONACYT, mucho trabajo institucional, como lo dijo la doctora Olivia, fue el primer QFB farmacéutico que fue por mucho tiempo jefe del departamento de orgánica, jefe de farmacia también, consejero técnico, y aquí muchas más actividades institucionales. Fue el que colocó, junto con el director en ese tiempo, Barnés, la primera piedra del edificio E que ahora habitamos. Ahí estoy, yo también, la doctora Isabel también, la doctora Raquel anda por ahí también, y ya vemos algunos que ya no nos reconocemos. El doctor Castillo, además de ser un académico excelente, un profesor magnífico, por aquí ayer o antier alguien se quiso retratar con él y dijo que era el mejor profesor que había tenido en su carrera, y también la vez que vino a la foto del departamento, la doctora Sobeida dijo que era el mejor profesor que había tenido en su vida, y lo cual es cierto, para mí, y yo creo que muchos coincidimos con eso. Pues siempre le gustó divertirse, vean allá arriba, dando clases de billar, dando clases de equitación, dando clases de natación, enseñó a nadar a Gabriel Navarrete, enseñó a nadar a Pedro Trejo, a Valdés, a Juanito Valdés, intentó enseñarme a mí, no lo logró, me la debe, doc. Entonces, a pescar a muchos, a comer bien y a escuchar a Pink Floyd. Entre sus muchos grupos preferidos, porque como le dijo a Isabel, no, aparte de la música clásica, que a él le encanta Pastorale de Beethoven, pues le gusta el rock clásico de Pink Floyd, otros más tranquilos como Camel, otros grupos como más modernos como James, etcétera. Aquí, el Cinema 122 nos hizo vivir viernes hermosos, porque cerrábamos las persianas, esperábamos que el sol se fuera, ya habíamos conseguido el queso, el pan y el vino, porque está prohibido beber en la facultad, ¿verdad?, en la universidad, y pues de pronto nos descosimos un poco y nos tomábamos nuestro vinito en esas sesiones de cine hermosas. El Conde de Montecristo, yo creo que lo vimos como 3 veces, Amadeus, una de sus películas preferidas, El ladrón de bicicletas, que cada vez que podía hablaba de ello, Malena, y la puse completa, doc, porque para verle las piernas, porque había otra que solo se veía su cara, pero era lo que más le impresionaba al doctor, las piernas de Malena. De esas películas, pues las vimos muchas otras, en las que me recuerdo una vez que estábamos viendo una película del jorobado con Gerard Depardieu, que no sé cuál era, en el que le hicieron una trastada los del pueblo a Gerard Depardieu en su papel de campesino, y terminó la película y se prendieron las luces y Jaime Pérez estaba llorando. Le había impresionado tanto, y entonces pues cosas muy, muy emotivas, bellas, que compartimos, aparte de la química, compartimos con él. El box es una de sus pasiones, y cuando fuimos a Filadelfia, en la biblioteca allá de Filadelfia, subió las escaleras como su ídolo Rocky, ta, 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 y subió corriendo, tararán, tararán, y entonces se quiso retratar con la estatua de Sylvester Stallone y como Rocky en ese lugar. Bueno, para terminar, él es así como me conocen a mí, que también digo muchas frases. El doctor tenía 2 muy importantes, y tiene 2. Deja que tus hechos hablen y no tus palabras. Algo que todos creo que hemos aprendido de una forma u otra, como dijera el Moreno. El problema es que todos somos diferentes, ¿verdad? Sin embargo, creo que eso es un legado muy, muy bonito. Deja que tus hechos hablen y no tus palabras. Y la que les dije el primer día de este simposio, no importa de qué esté hecho el violín, lo que importa es que suene. Si tú quieres buscar la cura o un tratamiento novedoso, una nueva molécula, no importa la metodología que uses, in cerebrum, in silico, intra lo que sea, utilizando lo que tienes en la gaveta, como empezó Gabriel Navarrete allá en Cuernavaca. Usaba todo lo que tenía en el gabinete y usaba su química, su diseño in cerebrum, y aquí lo tenemos también muy exitoso. Bueno, muchas gracias. Es un pequeño homenaje, pero también quiero, antes que nos aplauda, Doc, Fabián le mandó un saludo escrito que yo no voy a poder leer porque es muy emotivo. Entonces le pedí a un hombre que venga y lo lea. Venga, Carlitos, por favor. Y aunque lo escribió Fabián, nosotros pensamos que es el sentir de muchos de nosotros, o de todos nosotros.
Doc, de parte de Fabián, de mi parte y de todos los que estamos aquí, que somos de su familia. Querido doctor Castillo, es un honor estar aquí, aunque sea a través de estas palabras, para rendir homenaje a una persona excepcional como usted. Hace 21 años tuve la fortuna de ser estudiante de doctorado en el laboratorio 122. A lo largo de mi vida he conocido a muchas personas influyentes, pero pocas han dejado una huella tan profunda y duradera como usted. Recuerdo con claridad los días y noches en el laboratorio, rodeado de fracasos, fórmulas, seminarios, computadores, viajes a congresos, salidas de pesca, clases de fotografía y hasta de carpintería, y sobre todo, aquellos viernes de cine en petit comité. Pero sobre todo recuerdo esos momentos rodeados de su inquebrantable pasión por la ciencia y el conocimiento. Usted no solo nos enseñaba sobre química farmacéutica y diseño de fármacos, nos enseñaba a pensar críticamente y perseverar frente a los desafíos y a nunca dejar de cuestionar. Su enfoque riguroso y meticuloso hacia la investigación científica fue y sigue siendo una inspiración constante. Más allá de ser un mentor académico, usted fue un gran guía y un gran amigo. Su apoyo no se limitó al ámbito profesional, siempre tuvo tiempo para escucharnos, para ofrecer un consejo o simplemente para compartir una palabra de aliento. Esas pequeñas conversaciones en su oficina, en las que discutíamos tanto de ciencia como la vida, sobre música, cine, etc., son recuerdos que atesoro profundamente. Gracias a usted, descubrí el verdadero significado de la dedicación y el compromiso hacia la investigación. Su entusiasmo contagioso por cada nuevo proyecto, su paciencia infinita para enseñarnos y su capacidad para ver el potencial en cada uno de nosotros. Hoy, al mirar atrás, me doy cuenta de lo afortunado que fui por haber sido parte de su equipo de investigación. Las lecciones que aprendí bajo su tutela no solo me formaron como científico, sino también como persona. Sus valores, su integridad, su ética, pasión por el conocimiento son faros que continúan guiando mi carrera y mi vida. En este homenaje quiero expresar mi gratitud infinita por haberme permitido ser parte de su equipo, por su paciencia y generosidad, por haberme mostrado que la verdadera grandeza en la ciencia va acompañada de humildad y humanidad. Gracias por ser un mentor excepcional, un colega inspirador y sobre todo un amigo incondicional. Doc Castillo, su impacto en la química farmacéutica y en el diseño de fármacos en México es incuestionable, pero su impacto en las vidas de quienes hemos tenido la fortuna de aprender de usted es incalculable. Su legado perdurará no solo en las contribuciones académicas y científicas, sino en los corazones y mentes de todos los que hemos tenido el privilegio de llamarle maestro y amigo. Con profunda gratitud y admiración, gracias de todo corazón por todo lo que ha hecho y sigue haciendo. Una pequeña adición mío, espero que Fabián me lo permita y ustedes también, para que vean la gran calidad humana que tiene el doctor Castillo. En el año 1994, yo llegué de Guadalajara sin tener visa del CONACYT y con dinero limitado. El doctor se dio cuenta que a veces no traía ni para el camión. Me llevó a una de esas tardes a Perisur. Me ayudó a comprar una playera azul, un pantalón café claro y zapatos cafés. Claro, no son los mismos, aquellos eran talla 28, estos son talla 34, pero hice lo mejor posible. No crean que yo fui el único fachoso aquí sin saco y corbata y sin cinturón, pero un pequeño homenaje que recuérdase. Prometí que no iba.
Sí. Pues así como le compró a Charlie una muda, también se vistió a 2 que 3 en el laboratorio, les llevaba zapatos, les llevaba cinturones, camisas, chamarras y los invitaba a comer muy seguido. Creo que todos en su medida le hemos aprendido eso y nuestros alumnos pueden dar fe de ello. Hemos aprendido. Gracias, Doc. Y terminamos entonces ahora con este homenaje que ha sido muy emotivo para todos. Muchas gracias por todo, Doc. Lo extrañamos mucho.
Ok, obviamente son muchas emociones en este día para todos nosotros. Y vamos a proceder a la clausura de este evento como sucede normalmente. Primero que nada, pues les quiero agradecer a todos los presentes el hecho de compartir este sencillo homenaje para el doctor Castillo. Todos hemos sido, obviamente, instruidos en su humanidad, en su ciencia, en su academia, etc. Me voy a permitir ahora dar los números de este evento que normalmente sucede. Ok. Tuvimos la presencia, obviamente, de destacados investigadores, de exalumnos, todos ellos, en este caso fueron exalumnos del doctor Castillo, a los cuales les agradecemos su presencia en este evento. Algunos hicieron sacrificio, por decirlo de alguna manera. Creo que no es sacrificio, es un gusto estar aquí presentes. Les agradecemos a los ponentes, los nuevos o los pro investigadores, en este caso Gerardo y Diego. Les agradecemos a nuestras ilustres exponentes sobre sus experiencias con el doctor Castillo, obviamente. Y bueno, pues en este caso también hubo talleres impartidos, en este caso por los alumnos del doctor Luis Medina y por los alumnos del doctor Rodrigo, que siempre ha sido una parte importante en el desarrollo de este evento. Aquí hay varios trabajos que se presentaron, en total fueron 35, si no mal recuerdo. Y bueno, pues aquí está, ya me pasé, perdón. Vamos a dar los premios correspondientes. Una mención honorífica en el caso de los pósters de licenciatura para José Alfredo Guevara Torres, ¿está presente? Pásele, por favor, por su premio. Voy a ir optimizando un poco el tiempo porque creo que ya estamos cansados. El ganador en el área de licenciatura, Jimena García Vázquez, por favor, pase. Merece mención en maestría Valeria Jacqueline Soto Ontiveros. de premio es para Diana Evelyn Sosa Rojas. ¿Está presente? En doctorado, mención honorífica es para Ulises Martínez Ortega. Como ganador, Carlos Martínez Conde. Y aquí me voy a permitir hacer un breve paréntesis. El comité organizador de este evento anual ha decidido instituir el premio Dr. Rafael Castillo Bocanegra, que se va a entregar cada año en este simposio para los ponentes de licenciatura, de maestría y doctorado. Ese premio se va a instituir a partir de este momento. Bien merecido. No sabemos si va a ser honorífico o con algo monetario todavía. Ok, en este caso, ahora tuvimos la participación de 98 a 117 participantes, hubo en carteles de 50 a 60 participantes y en el curso taller de 18 a 24 participantes. Y las instituciones que nos acompañaron aquí están listadas, cabe destacar, pues obviamente, Medicina, UNAM, Zaragoza, Cuautitlán, etcétera, ¿no? No quiero continuar con todas, pero lo que nos da mucho gusto es que haya instituciones de provincia que estén participando en este evento, ¿no? Por otra parte, aquí tenemos las visitas que tuvo esto, porque esto también se proyectó a través de YouTube, y como podemos apreciar, tuvimos bastante audiencia, y sobre todo en la parte del continente. Y en otros años creo que ha habido más participación de Sudamérica, si no mal recuerdo, pero en esta ocasión hubo de otras partes, así como en la parte de Europa. Ok, tuvimos nuevos visitantes, algunas sesiones por dispositivo, o sea, las visitas que tuvimos por dispositivo. Y bueno, en este caso quiero destacar la colaboración del doctor Francisco Hernández, de la doctora Alicia Hernández, del doctor José Luis Medina y del doctor Rodrigo Aguayo, para todos ellos mi más sincero reconocimiento por el apoyo que nos dan, pero en particular, me voy a permitir solicitar un aplauso para el doctor Rodrigo Aguayo por su entusiasta participación con todos sus colaboradores. Cabe mencionar que también dentro de esta organización está el doctor Gabriel Navarrete, el doctor Carlos Méndez y obviamente el doctor Antonio Romo. Ellos son integrantes del colegio que le llamamos nosotros de Química Farmacéutica, todavía no oficial, pero siempre nos han apoyado con su destacada colaboración. Aquí, gracias. Aquí están los chavos que han estado detrás bambalinas, haciendo el registro, llevando la asistencia, etcétera, moviéndose para los bocadillos, pegando los pósters, moviendo las arañas, etcétera. Realmente ellos son las hormiguitas que nos han ayudado a que este evento siempre sea un éxito cada año. Por favor, un aplauso para todos ellos. Bueno, y siendo las 14:52 del 14 de junio del presente año, se declaran clausurados los trabajos del presente simposio, 10º Simposio Tendencias Actuales en la Búsqueda y Desarrollo de Fármacos. Muchas gracias por su asistencia. Buenas tardes. Goya, Universidad. ¿Onta? Ahí lo viene.